It's okay. Yeah, I have water, coffee. I'll get it. Just I'll keep it here. Okay. Good morning. Assalamu alaikum, everybody. So let's start the day four. Day four, right? Day four of the drug discovery course, multidisciplinary approaches towards drug discovery. So today's my topic is I have two topics today. This one is kind of okay. I think yeah, that's better. So टॉपिक जो है वो आज एक तो ये है ड्रग डिस्कवरी इन एकेडमिक पर्सपेक्टिव और दूसरा इंटरैक्टिव सेशन. सो इंटरैक्टिव सेशन आई वाज थिंकिंग इन अ डिफरेंट वे. वी विल डू दैट, बट देन लास्ट नाइट आई टॉक्ड एंड आई थिंक आई एम गोइंग टू गिव short presentation of some of my work, and then we will make some group, or I will ask all three instructors to come, and then you guys can pose questions. So this will be a better way if they agree with me. So about the academic perspective, this may be a controversial topic for us today, but let's see how it goes. I come from industry and academic both, and other people are from industry and academic both. So let's see who has what. So please come. So this is a statement made by Tim Hammonds, who was the senior vice president of drug discovery, and the problem mostly is the funding in case of academics, and the industry has a lot of money. So academics don't have funds, and pharma cannot risk funding the project of the failure. So pharma thinks that all project which we do in academic is a fail, okay? And academics think that pharma is not doing anything, they are just making money, right? Which is in some way it is true. So I have seen both angles. I'll try to, go through a little bit and then pose some questions what you think about it. So remember this statement, okay, that academics do not have funds and pharma cannot risk funding project failures, okay? And this statement is given by a very famous scientist, Tim Hammonds, who discovered several drugs, and he was vice president at AstraZeneca. So my outline will be why drug discovery, why we should do drug discovery in academia, okay? You just get the degree and go and work somewhere as a professor, as a instructor, as a engineer. Why we do that? So now, challenges of drug discovery in academia. Funding is a major issue for academia. In industry, I used to order a chemical or a compound or a device, and the timeline is 48 hours maximum. Wherever it is in the world, it will come to you. Especially for big pharma, Bayer, GlaxoSmithKline, Lundbeck, where I worked, Bristol Myers Squibb, it was like that. Just it will come to you, and next day it will be on your bench or your table or in your office. What is the recipe for success if we are in academia? We talked about it, how much academia has given to industry. Maybe I will say from industry that we are ungrateful, okay? We are really ungrateful. We don't care about academics. 80% of drug, I will show some of the graph maybe later, that 80% of drugs have origin from academia, okay? Or it was discovered also in an industry. I'm not saying they are totally useless, but origin is from academia. So basic research industry don't do. Basic research is done by Dr. Azhar usually, and then we pick up from their literature and we do that. Future opportunities and my final thoughts. Okay, so history of academic drug discovery. Academic drug discovery centers, ADDC, remember that, there are many now. So what happened that industry, they started exploiting academic institution So academic institutions, they started making their own consortium. I talked about Emory University, if you remember, and there are many more, and I will list some of them and you will see that. So academic bioscience research started sometime in 19th century, okay? And early 20th century, the NIH, which was National Institute of Health, was established in US to effort in biosciences research, and they are the most funding company, most funding agency to give funds to academic institution. In 1970, 1980, a spin out from universities resulted in biotech industry, and I mentioned that Genentech was the first biotech industry. It is now valued at $120 billion. It is almost 50 kilometers where I live in US. And yesterday we had a old friend from Genentech, and Dr. Azhar showed me his message, and he is one of the founder of the company which we launched, and he's friend of him also, alhamdulillah. Center for Academic Drug Discovery was recorded in 1980. Florida was the first center which were recorded. So I'll tell you why Florida. Everyone knows about the cancer drug, what is, just slipped my mind. That was actually that was synthesized by University of Florida. The professor, his name was Horton. So he synthesized that. And they got a lot of money from Bristol Myers Squibb, and they built a whole new building for that. And at the same time, Emory University was also working when HIV hit the world, and I will show some name of the drugs, which was done by Dennis Liotta and Raymond Shinazi. Their photograph is there. So University of Florida started drug discovery center because it is an academic center, and they never thought that they can do that, because in universities, in academics, a professor want to get the grant, they want to publish, okay, and they get the promotion and some awards. But financial award was not there. Financial award was all taken by industry, okay. So now the thing has changed. Initially, organizations were operating under loose collaboration. For example, I am a professor in Aligarh Muslim University, and I contact Zydus Cadila that, okay, I will do some work for you, or I will discover a disease target and I will give to you and you do that. So this was kind of arrangement earlier. Now it is not like that. Everyone is holding on to their inventions. Integrated group under one management started, for example, Research Triangle Park in North Carolina and University of Dundee in UK. Why I put these two places? Because I know people who are working there, and University of Dundee, my very good friend Mahmoud Ahmad is the head of the drug discovery. We work together at GSK. As a recent count, there are about 76 ADDC, means Academic Drug Discovery Consortium or centers in US. In Europe, there are 15. Even in Middle East, there are 4, mostly in Egypt and Jordan, not in Saudi Arabia. Australia has 3, and there may be many more. I don't know. In India, also CSIR. I was one of the awardee of JRF and SRF, okay? And we did work there. I used to go, when I was at IIT Kanpur, I used to go to CDRI a lot, and I used to come to Delhi also for. So CSIR is another institution which is now promoting academic drug discovery to the industry. So remember all those things, okay? So university and government laboratories are becoming credible place for innovation at early stages. I mentioned before, and my other colleagues, they mentioned, academic institutions don't have that much money. They don't have deep pockets. They don't have deep pocket to spend that much money in development. So what they do, they do initial work, and that's very important actually. sometimes it is not paying, but it is very important. That is the foundation of a medicine for a disease. What academic institutions do that, and we have to admire that. Like Dr. Azhar, he has, he found out about 10 genes in colorectal cancer. Work is still going on that, and hopefully one day there will be a medicine for that. So this is how it works. Myself, I have discovered many drugs, and most of my compounds, which are not even drugs, they are sold by I didn't have the list, I forgot to bring. There are 17 compounds which I made in my career. They are already sold by companies as a tool compound, what we call it, okay? So they are used for research purpose. They are all patented, and I see that even I have to buy those compounds. Recently I bought, for Noran, I bought 50 milligram for 6,000 reals. So when you buy your own compound, you feel bad, but you feel very elated. Okay, I made this compound in my life, and which is sold by some company. Okay, so the individual freedom, intellectual diversity, flexibility, and originality. This is the hallmark of the academic institution. They discover, they do the original work, which other people don't touch it. Okay, so that's important also. And they have freedom. If they fail, so what? But in industry, if you fail, either you will be fired or you will be demoted. Intellectual diversity, we have in academic institution, we have thinking, plain thinking. We can think anything out of the box. In industry, you are just focusing on a narrow lane. Make this thing, work on this thing, make money, and move on. That's what industry is. Now, flexibility and originality is the hallmark of our academic institutions. They have expertise, and our academic institution, they have expertise in basic biology, which we don't have in industry, medical, chemical, computational, and mathematical sciences. Several times I mentioned industry don't do all those things. Okay, so we get lot of advantage from industry, from the academic. That's remember always that. So you are in an academic institution like AMU, you are serving humanity, I will say that. You are really doing good work. Industry, but now things are changing, which I'm going to explain in few more slides, that you have to protect your rights. That's important now, okay? A small investment, then incremental investment of these laboratories will bring enormous added value to institution. So now the themes are changing. So people are smart now in academic institution. They ask industry, because I'm talking about academic perspective, so I'm talking from you only. So academic institution, they ask, hey, industry, Zydus Cadila Cipla, why don't you invest into us? So we will do some work for you, and then we will share the bounty of that. Sometimes they do that, sometimes they do a big investment. We have done recently in Saudi Arabia, we are academic institution right now. We have done investment in Flashpoint. There is a company called Flashpoint for nanotechnology from Northwestern University, and we invested 50 million with them. We are building a mirror lab like they have in Northwestern University. We are building a same lab in Kmark, and we will do the same kind of research. That's how it goes. The second agreement is done with AstraZeneca in Sweden, and there are 5 or 6 people there right now. Okay. So see, this was not there before. Okay. You cannot ask industry to invite you or invest in you. Now things have changed. They know that if we don't get things from them, we will not survive. Okay, drug discovery funded, and so this is the industry. But for us here in AMU, we have to go to charitable organization also. And I don't know about India, what is the situation here, but there are funding organizations like Bill Gates Foundation is there, Fox Foundation, and Evolution Foundation. There are many more foundations in US. Recently, I'm talking to a foundation which is called MMF, Multiple Myeloma Research Foundation. It is based in San Francisco, and Stem Cell Foundation is in US. So there are many foundations. So they are also funding. So they are charitable foundations. So they are all funding research.
In India also, like the Jubilant itself, they do have a, because some of the part which goes in the CSR, industry. In a similar way, we have a outspin. We have some funds for the people who want to do some startup with the budding ideas.
Yeah.
We also fund from a Jubilant.
Okay.
Similar line, there are many people. What we are seeing in these days, presently, people from US who have been well off now, they have been angel investor for many of these.
Yes. Yeah. Yeah.
That is what, just to add.
Angel investors, actually, yesterday I talked about angel investors are people who have, they are private people. They don't have to do any research. For example, in some cases, somebody's parent or father or mother or sister and they have big money and they died, unfortunately, due to some disease. Fox Foundation is one of the example. Because he had this disease, so he made the Fox Foundation. So similarly, angel investors are there, and this kind of culture has to be developed in India and Saudi Arabia also. So we started talking to people who have really big amount of money sitting somewhere doing nothing, so they should start investing in research. And there's a organization called Sanad in Saudi Arabia. They started funding glioblastoma, a cancer of brain. So one of our friend, he got about 300,000, $3 lakh from them to do some research. So this is another thing that we have to think about, that how we can get money from the angel investors, venture capitalists, and charitable foundations. A very good friend of mine here in AMU, Dr. Sohail Sabir, he has given 50,000, 50 lakhs, right, sir? 50 lakhs, he has given 50 lakhs. He was a teacher for 30 years in chemistry department. He has given 50 lakhs to the university to invest on you like a student, give the scholarship. And I'm working with, although I'm not a Aligarhian, but I'm working with Aligarh Alumni Association in San Francisco from 1996. We do a lot of, charitable work, and we get money and fund a student. Now, actually, it will be a good idea for Dr. Azhar to talk to Alumni Association to fund research also. They are funding almost 100 students per year. Not only here, someone who is very smart, they can fund there also. So this also happened recently, it started. So I think it is time for Alumni Association to fund research. And then there's a government route will address unmet medical need for a small patient population. You remember yesterday we talked about that how we are going to fund a precision medicine. Somebody asked about personalized medicine. This is a small population of patient which government is not going to spend any money, industry is not going to spend any money. Who will spend? Me and you, okay? Or the Alumni Association or charitable foundations. Okay, this is, I think it's not very clear. It is in, I will try to read from my screen. Let's see, how can I go to full screen? How can I go to full screen? Anybody can help me. I'll just take a so that I can read. Yes, okay. I'll go back to the presentation mode. Okay. Yeah, yeah. Okay. So yeah, that's what I was forgetting the name of the drug. So anybody knows about Taxol story? Taxol story? No?
I know the Abraxane story, that's what is very
Yeah.
famous.
Why I'm asking that there was an Indian involved in the isolation of Taxol, Mukesh Wani and the Robert Wood. There are two people. Taxol is a cancer compound which is isolated from the Pacific U, where I live. But if you keep on isolating from Pacific U, all the trees are gone. So after isolation, initial work was done on the Taxol. Then chemists came and they started synthesizing so that they can synthesize and give it to the patient. So this is a target which is a microtubule stabilizer. And Mukesh Wani was working at Research Triangle Park, and this was licensed by my previous employer, Bristol Myers Squibb, and they gave about $350 million to Research Triangle Park, and of course, part of that Mukesh and the other person got. So this is like came from from the academic, and academic gained from it. Especially there are other drugs are given here. You can see that. So these are all academic drugs. They came from academics. Wodinstat or Saha, this is stone deacylase oncology. It was discovered at Sloan Kettering Research Center and licensed by Merck. Okay. For example, cisplatin or paraplatin, you call it. This is a DNA very, very well-known DNA alkylating agent used everywhere in the world. It was an Institute of Cancer at Research, UK it was, and again licensed by Bristol Myers Squibb, and they got a lot of money. This is for example, another drug, methyl alkylation oncology drug, that was discovered at Aston University, UK, sharing flow. They paid them hefty amount of money. This is another drug, cardiovascular drug, which was developed at UK, and it was given to Pfizer. Similarly, Alimata, that was another dihydrofolate reductase inhibitor, and that was developed at Princeton University. So I just want to show you that how much work our academic institution has done, and and every drug is billion-dollar selling drug, okay? So you can understand that what is the value of okay, yeah, this is called AZT. Everyone knows that it revolutionized the patient, health because it was a reverse transcriptase inhibitor. And this was discovered at Michigan Cancer Foundation, and it was given to GSK. And then this was given to all over the Africa. It saved millions of life, and it was made very, very cheap by Gates Foundation. Okay. Can I have again full? Then after that, this is the last one, and then I will have the. Just so that everyone can read. Okay, I think that will be good enough. So these are what I showed so far was the old drugs. They are already on market. Uh, then there are new ADCs come up, and this is a caliber at Scripps Institute. They are working on several, and you can see in the last line which stage they are. You will have these slides and you will see that. So it is in phase 3 clinical trial. The other one is at University of Pennsylvania, Center for Cellular Immunotherapy. Chimera is a very well-known medicine right now, CAR T-cell. You heard about CAR T-cell yesterday. It is for B-cell lymphomas, and this is already FDA approved. So this is the second gene therapy which was approved, fully discovered at a academic institution. Okay? And this kind of work industry cannot do. I'm telling you, I spent half of my life, 2/3 of my life in industry, and they cannot discover that. Emory University, I want to talk about that because they were the first one to start a consortium kind of thing, and they were very smart. They already had the special people who know how to deal with the industry. Attorneys, experts, key opinion leaders, and they discovered several drugs, and one of them was Epivar. MTVAR is another one, and they were HIV and HBV, and the person who discovered his photo I am going to show you in next few slides. Maybe it is here. So they are FDA approved. And these drugs, they changed the life of the patients, and it was given at a very cheap price to the, especially in the Africa. I'm sure it in India also, and it was all synthesized and manufactured by Cipla here. And they licensed it. So, but they got a lot of money out of it. There was a litigation between them and so NIH because the work was funded by NIH. This is another important area to understand. The research work was funded by a grant from NIH. So they should have a pie also from the everything. So they fought, Emory University fought with them, but Emory won because the idea was there. The funding was done by NIH. So why they should get, their job is to fund research. So Emory won in the US Supreme Court. So this is one, just I wanted to make sure that you understand the dynamics of these discoveries also. There is a high throughput screening center. So if you make a compound or antibody, they will do for you free of charge. Similarly, NCI, National Cancer Institute, they can test your compound free of charge. Okay, Warren Center for Neuroscience and Drug Discovery. So my point is that every area of research, there are some institutions who are doing that, okay, academic institution. So you can see that there's a drug already in phase 1 trial of Alzheimer, and somebody was talking about dementia, so maybe you can get in touch with them and talk to them about what they are doing, and maybe they will help you. So everybody who has that some idea, it is there. Okay, yeah. So funding is a major issue for academic institution. Successful ADC should have multiple funding streams, not, I already mentioned not only NIH, NSF, or Department of Defense. This is about US or in Saudi Arabia, we have Saudi NIH, SNIH, and Research Development Innovation Authority and other institutions which are government institutions. Funding with disease-focused foundation or entities like I already mentioned, Bill and Melinda Gates Foundation, Tropical Disease Foundation, Rare Disease Foundation, King Salman Foundation for Disability. So like tropical disease and rare disease, nobody want to fund it. So you have to find someone who can fund these things. Only yesterday we were talking about AMR. So after cancer, AMR is the most difficult thing to treat, and we don't have anybody to fund that. Actually, in last 30 years, first antibiotic which was discovered by Wokhard, in India, nephtharamycin, first time. Okay, in 30 years, all big companies, they didn't discover any antibiotic. All you are taking is first generation, second generation penicillin or whatever. Even tetracycline is coming back now. I was talking to a physician in Saudi Arabia. Partnership with disease-specific pharmaceutical, it is very important for academic. My friends from academic, they have to partner with the pharmaceutical company. Partnership with disease-specific philanthropic organizations, Fox Foundation, and funding with angel investors. Dr. Zain just was telling, so I said that I have a slide for that. You have to find who can fund your research, and there are people who have money and they will fund. And we are trying everywhere in the world and in US also. Recipe for success, focus on commercial potential with a strategic project. So when you design a project, think about it that you are not going to get only paper, you are going to get a product, okay? So that's important. Then people will fund you because they will see the money, okay? He's talking about something which will make money for us, but your job is, you are very nice that you are going to do, you are thinking about funding, you are thinking about treating a patient, but they think about money. So this is a dynamics you have to use. Focus project with unique expertise. So this is also important. Not everything is important. You have to think about really niche project, okay? Think about, from any, I'm not talking about from microbiology or any other perspective or chemistry perspective. You can think about a device which can detect blood sugar or hemoglobin A1C. There are devices there already, people made. There was a, Google has made some kind of device you put on the eye and it will discover how much blood you have. People have their sensors. Maybe simpler. There are many outside, but you can simplify it and make it cheaper. That's the important part. Focus project with unique expertise, niche specialization. I already talked about the unmet need. There are the specialized, very few people are there, so you can work on that. Build in-house comprehensive drug development infrastructure, and that is for the administration here, that they should build an infrastructure. I was so happy to see Dr. Azhar's lab, that he has almost what he's doing. So please be in touch with him. He might be helpful on your projects. Regional ecosystem, that means that in UP, for example, you have to find people who are really interested. I can give you some names. CDRI is there. There is an institute in Roorkee, I think. They are doing, because I'm looking at the literature and I know that who are really doing good work. And Toxicological Research Institute in Lucknow, that is also there. And there may be many more. I may be missing some of them. You have to get in touch with them and make a consortium with them. So this is regional ecosystem. If you go, from Aligarh, if you go to Kerala, it is too far. It may work. The SAMI lab is there, for example. They can fund out. If you go to Himalaya, which is in Bangalore based, they will do that. But regional ecosystem is also very important. Within university, outside university, between two university, between industry. Those kind of things can be done. Okay. Integration of regulatory affairs, project management, clinical development, fundraising, and market analysis in ADCC, all has to be done by the administration of the university, so that the researchers are free of doing that. Most important, adoption of emergency technology with ADC. So emerging technology. So I talked about yesterday, 17 of them. So pick one and think about it whether you can do it or not. That's important. I keep on emphasizing whether you can do it or not. Okay, let's some stories here. Emory of successful academic drug discovery. These two people are far apart. They don't have any connection in this science. Dennis Liotta is a hardcore organic chemist like me. Raymond Shinazi is a pediatrician. They got together and they developed several drugs. He's a virologist. Raymond Shinazi is a virologist. They developed 3 HIV drugs. and which saves millions of lives. They both have collaborated. They were within academic collaboration. Raymond was courageous to set up first HIV lab to test compound. So when HIV virus and now MERS, SARS virus hit, it was not easy to set up an assay lab because this is called BSL-3 lab or biosafety level 3 lab. Not everybody can work. They have to be specialized, trained in that area. But he was, and at that time, forget about that. This was like 30, 40 years ago, right? 1980s it hit. So he was courageous to work with a live virus. Live virus means he can be infected also at the same time. There was no drug. What happened to SARS? People started doing, they were infected, but we didn't have any drug, right? So this person took risk of his life to set up the lab. Sometime academics, they do that. In industry, nobody will do that, I can assure you. Their collaboration resulted in Epivir and Emteravir medication used by 400 million people. This may be old data, maybe 1 billion people, I will say, okay? And it resulted in largest deal ever for a academic institution, 525 million, $500 million royalty for Emteravir, and large portion was reinvested in research lab. So Dennis Liotta, he is a very honest person. He didn't take away all money, or Raymond also. He didn't take away all money. They invested, they built a whole new building in Emory University. You can go and see that. It is with their name. Second is Pittsburgh story. This is a quest for amyloid imaging. I think people were talking about that Alzheimer. So Dr. Chester Mathis, he was a radiochemist, and William Clark is a psychiatrist. No connection at all between them. A chemist, a psychiatrist, okay? He was an MD psychiatrist. No connection at all. They got together. They worked together to create first PET tracer molecule to diagnose Alzheimer's disease. PET tracer molecule, anybody remember I mentioned in my first talk also? What is the meaning of PET? Perfect, very good, thank you. So that is a radioactive molecule which is injected, and it will light up where it will bind to the disease area, okay? So this is called PET tracer, and SPECT is also there. They emit gamma emission. PET tracer, they emit gamma emission. There is a radioactive counter there that will measure, okay, where it binds. So first PET tracer diagnose Alzheimer. This molecule crosses blood-brain barrier and stick to beta amyloid. So you know that Alzheimer's disease is a disease of brain. So the molecule has to cross the blood-brain barrier. I am not going to talk about that. This is another big problem for chemists. You have to make a molecule that crosses the blood-brain barrier. See how nature has protected your brain. Not everything goes inside your brain, okay? So that's the thing but you have to make that to work like that. The score is there, and they provide the severity of that. I think I have 2-3 more slides to go. So reverse, and there is something called reverse academic collaboration. And you have to go in the niche area, okay? And real-time interaction should be important. So this is very unusual in academic. I will give you the example who did that. This is very unusual help from academic to the industry. Usually biology is from academia institution, chemistry from pharma industry, right? Every medicinal chemist identified a trisubstituted cyclopropane cystic fibrosis. This is the very latest, just last month it was published in JMET Chem. So they discovered this molecule from their library, and they made a small molecule, but they didn't know how to make large scale. So now, this AbbVie or Abbott is a big company. They are medicinal chemists. They didn't know how to make it in large scale. This was very nice. So this is, we call it a reverse academic collaboration. So they came to Emory University. This was a challenging chemical space to develop the drug, and AbbVie took help from Davis Group in Emory University. he asked Davis that can you scale it up from milligram to 5 kilogram, for example. This rarely happens, so this was one of the case. A flow process was developed, which you can see here. I'm not going to go into detail. When you get to the slides, you will know that. So collaboration accelerated unique target synthesis. So this is a very complicated thing they did, but actually it is very simple. They used some kind of flow, we call it a flow syringe, and they started doing that, and they could make intermediate, which was very difficult to make, which is last in orange color. Okay, so this was all done at the academic institution, not in the industry. Okay, industry could not do that. So 50 gram compound fit for purpose was delivered to them. Then kilogram synthesis they have done in a flow chemistry system, and finally they made the project and lines. And of course, Abbott was very happy with that, and they paid them a lot of money. Okay, how academic and pharma can work together. So this is a chart. If you see that where and how they can work. So this is academic institution. They do fundamental research, right? And then presentation, publication is our hallmark. We want to do that. And then position and promotion and funding. That's what we are looking for. But we have to look a bigger picture in academic institution also, which is that you think about the clinical and clinical candidate and clinical trial. You have to talk to clinical trial expert, and Dr. Azhar told me that we are going to talk some clinician tonight, and this is what it is, that the academic institution is talking to clinician, and the pharma and biotech will take your patent, your invention, and they will, of course, they will pay you, but they will develop it because we don't have the resources to develop. So this is what happens, that pharma industry takes your invention and then they test, confirm, then they go to patient validation, efficacy, safety, and all those. Yesterday, Dr. Zain talked about it a lot. Business development decision is made by company because they are looking for money, okay? And then human proof of concept, which will involve both academic and the institution and the industry, and then finally we get a target, hopefully. So this is the chart how it should work. So future opportunity for academic and drug. Let me see. Okay, I'm on time. The era of dominance of big pharma is waning. Why I am saying waning? Because they are not doing basic research, and you are doing basic research only. Okay, so the era of that is slowly it is going down. Cost of development of drug is skyrocketing. So you need to do, academic institution need to do a lot of research. Legislative pressures are there. You heard Trump is asking everybody to reduce the price of the drug, and in India also I heard that government organization and universities are emerging as a key player in drug discovery. So your future is very bright. That's what I will say. In US, NIH funding has become credible source of novel mechanism and health diseases. Many things came from NIH funding. This was later exploited by for innovative medicines, for academics, rare disease. This is my advice. Rare disease, tropical disease, neglected disease, some infectious disease will be a gold mine to work on. So you can think about it. Of course, you have to work on cancer and cardiovascular, but this area can pay you, and it can discover something new. Academics should work on public-private organization like Medicine for Malaria, MMV. This is another organization, and a drug for neglected disease, DI. The future of ADCG will be based on two factors. One is funding source, other is strategies to drive translational drug discovery. Translational drug discovery, we talked about it. We can discuss more. That is my last slide, and any questions? So I'm 45 minutes now. Okay. Any questions? I know it was a little bit, different, kind of boring also, but this is what I gave you an idea how important you are in this research in drug discovery. Okay, and drug discovery is going through. Now it will go through the academic institutions. Okay, thank you so much. I think I will let Dr. Zain to talk about his work.
No questions, I'm surprised.
Yeah, I'm surprised too. Looks like everybody is sleeping, so they need another coffee or 5-minute break.
Yeah, we can do that.
Yeah.
Don't go outside.
Yeah, don't go outside. That's right. Yeah. Yeah, somebody was asking. Yeah.
Hello.
Mosquito related, malaria.
Malaria and several other.
Yeah, there are drugs, artemisinin, chloroquine. There are one more I don't remember. It's stored also in academic institutions. Okay. And chloroquine are also stored in academic institutions.
Thank you. Yeah. So good morning, assalamu alaikum, and glad to be here once again. Is it possible for you all guys to come forward? Hey, come forward. It's good. It's the best way to the people sitting front. They can go back, move yourself, get yourself a stretch. Come forward. Come forward. We all want to see you guys. You have spent so much time, and I want you to see closer to me. Come forward. Yeah, sit. We are not going to bite you, okay? Sit. Come forward. Yeah, this is. हाँ नहीं मैं. So we are on job now, okay? Yeah, so just before we start, I want to give you a recap. It's been a fascinating 3 days. We are on the 4th day probably, and hopefully you would have got something to know about drug discovery and development. And I just want to recap from the very first day. You understood that what the biology is, what is the different stages of drug discovery. We talk of it. We talk of target identification. Yesterday it was touched more of a cancer, the biology, the disease, cancer as a biology disease. We all have learned something more about it. Then we understood the two arms of drug discovery. One was the efficacy or activity, which we say. Another arm, how to make that compound which should be an active. And what is the role of a chemist who is a who is the taskmaster in that whole process, and who takes care of all, who is the center stage of it, how he tweak the compound, how we make the baby, and baby becomes the boy, and it graduate, and it's all become one of the thing which we all know well. It's saving a life for millions and billions. Along with that, in industry, sometimes it has been quoted, one of the people who are the beneficiary of a system, they take a lot of money out of it and they earn money, but there is a lot of investment. We have seen there is a 2.8 billion as sum with total of a money which is spent. But the question is, why the hell you will spend so much money if you don't get something? I have already told the Ketroda story that it makes money every year what he spent for the earn for the whole month, spent for the whole drug development. So there is a balance between it. Then we talk of a lot of the academia, role of academia, how the collaboration with industry and academia will be helpful to you guys, and that's important. Academia is the most important arm. Most of the drugs which has been there, I would have shown you a list, or sir has already told you a list, that how much the role of academia has been in discovery. You guys do the basic biology, basic discovery from the academia, and that's what help us to take the leaf out of it, and we as the industry try to tap on it, and the chemists work on it, and we all do it. So important is, there is a lot of things, there is a huge flow of information has been. I probably feel that you guys have taken some out of it, and I don't want that you all take all, but choose the thing which interests you, and hopefully tomorrow you guys can be a better researcher. You might have an aim to do anything, and that is the only aim which we have here. Okay? So take one leaf out of it, that I want to do this research, I want to go to the industry, I want to do my future, and I don't want to be a very hi-fi scientist. No, we need a small stuff, and I think that will make a huge difference. Okay? So now the question started, how we build up. The thing is, what is the potential pitfalls and alternative strategy in a drug discovery? The task is given to me to complete what exactly is. So I will try to cover some of the stuff before we go. So this is what the central dogma for a drug discovery is. You all know well central dogma. You know the DNA, you know the transcription, translation, we talk of a genetic term, but for a drug discovery this is how it is. You have a target selection, you lead discovery, medicinal chemistry support, and then in vitro studies, in vivo studies, and then you have a lot of, these are the preclinical, and then the clinical trials and all that goes forward. This is what you already run. Hopefully you all have remembered this slide many times. This is how we feel, you know. You all talk of telling that industry being a selfish beast altogether, people take off it. No, that's not the case. We spend money, we do a lot of stuff, and we all are also human beings. So there is an emotional touch to all of us. We all do science, and once the experiment fails, we all get crazy. And not one person. I don't remember in my early days when I joined 2004 industry, and till 2008 I used to come home by 6. We came to at 11, 12 sometime. System stopped, we have to develop these timelines and all. It's a very tough place, you know. Industry says, you work for me, I will give everything to you. I will take care of food, coffees, stay, putting up here, going there. You don't have to worry. And you don't have to run for any chemical, you don't have to arrange anything. There is a call there at a talk, you have to talk to supply chain, hey, give me this molecule. That's their headache, nothing you. You don't have to run, you have to work and focus on your work, that's all. And probably that's the biggest difference which academia and industry have, the resource and how you do get it. So this is what I want to bring, I wanted to make an image of it, just to say you 2.6 billion, there is a time ticking to it, discovery and expensive and time consuming, and it took 10 to 15 years, we all talking on these things, and then all of the sudden you feel this is an expensive, exhausting and essential mean, you know well, but what happened where hope meets a hard reality, and that's the price of discovery. It's not the money, it's the this scientist gets emotional how he tear his hair just once he gets the failure in front of it. This is a slide which tells when promise meets reality, real world fallout. So I'm giving you some example. This is Vioxx, if you see, this is a financial impact. See the financial impact. This is 4.85 billion settlement and then $2.5 billion lost sale. It happens to a company, and it was the simple reason was the cardiovascular tox. It was pulled out. Similarly, if you see Avendia, increased risk of heart attack, and this is the money which was involved. So what I want to say, you develop a drug, in the process there is a failure, fine, we all understand, and we can rectify, we can go back to the board, chemistry can go back to the board, they start making a new molecule, they will design a new molecule and provide the thing. You have a failure, fine, this is taken. But sometime failure happens when you go to the clinic and there is a withdrawal. And that withdrawal, why it happened, I am giving you some example. These are the withdrawal, failed efficacy despite acceptable safety. These are the, and there is a cost reported. Yeah, I know you guys have been seeing the newspaper of late. There is a story with the cough syrup going on here. It was exported and India causes a child death and all. That's also it was a ignorance, okay? It was a regulatory aspect which was not taken care of it. Fine. So what I want to make, these are the small pitfalls but have a huge impact in terms of a timeline, in terms of your own image, in terms of the image to the industry, to the people who are taking the drug, and this is the financial impact, which is a great stuff for any business running places. So I will take one by one how the disconnect in the drug discovery happen. There is a drug, if you see the disconnect, there is a fragment. The biggest problem we all talking from last 4 days, I think if you can undermine, or if you can just focus on it. If I ask you, give me 2 words which you have been repeatedly listening throughout 4 days, is one is the collaboration. You are listening this thing, and that's a reason for many failure. Most of the research happened. I was in one conference and there was a guy who was Eli Lilly CEO, and he was speaking a lot on AI, he was talking of these things. But the problem is none of these pharma people put any failure outside in the market. They all do a siloed research. So similar happen in your academic. One lab doesn't know what the other lab is doing. If you are, that's a small chemistry reaction, somebody knows it, and if fails, it doesn't tell I failed when I did this. If they tell everybody, people will not try it. They will do something different. So it's important that we should be transparent, and for transparency you need a confidence. The biggest problem I feel sometimes is you do not confident of. Industry has a different thing, there is the IPR, there is the money associated. Academia, if they remove the barriers, things can change. Similarly, in discovery setup, there is a siloed drug discovery, absence of feedback loops, that's what I am trying to, it's a missing communication. What happens? Try to understand the flow. Chemists make a molecule, there is a target identification, everybody decide that we will work on this target. Then you ask the biology that whether you have an assay to test or not, fine. Then we ask chemists, can we target this protein? This is how the protein structure is, can we target? Chemistry start working. Then we take a computational help to tell about the structure and how the flow of in silico biology looks like, or in silico chemistry looks like. Then it goes to the ADME. I don't want to go further. There is a continuous flow of information between a chemist and a biologist. Chemist tell, take this compound, I made it, you just see the structure, they say, and give them the activity back. And then there is a discussion. थोड़ा सिग्नल दिख रहा है, अगर तुम थोड़ा चेंज कर दो तो ये पोटेंट हो सकता है. अगर ये कम्युनिकेशन ना हो और सीधे डाटा भेज दिया जाए, और वो केमिस्ट को उतनी बायोलॉजी की नॉलेज अगर नहीं है, तो वो कहेगा या फेल हो गया. वो बिल्कुल हो सकता है तीसरे डायरेक्शन में आ जाए. अब जैसे हमको घर से यहाँ पहुँचने में हमको इंजीनियरिंग कॉलेज आना है, अगर कोई हम बावे सैयद पे भी खड़े हैं और किसी ने मुझसे कह दिया कि यार तुम एक काम करो दाहिने हाथ पे मुड़ जाना तो उधर की तरफ निकल, तो हम तो जकरिया की तरफ चले जाएंगे, हम इधर की तरफ नहीं आएंगे। हैं कोई बता दे बहुत सही पहुंच गए, बस थोड़ा सा वहीं से नेविगेट कर लो, रास्ते में तुम थोड़ा सा आगे जाओगे तो दाहिने हाथ पे चले जाओगे, इंजीनियरिंग कॉलेज पहुंच जाओगे, नहीं तो वो घूमता रहेगा। ठीक है ना। सिमिलर इज द केस इज द फीडबैक लूप आई एम ट्राइंग टू टेल यू। देयर इज अ कम्युनिकेशन which is important, there is a flow of information and there is a constructive suggestion, and for suggestion there is a taker also which should be there. Then you have impact on the development efficiency, risk of a, so this when you don't have efficient you waste a resource, efficiency नहीं मिलती है अगर ड्रग की तो तुम्हारी रिसोर्स भी खराब होता है, डिले भी होता है और ओवरऑल सक्सेस पे डायरेक्ट वेस्ट होता है। Risk of late assessment, this is something very important. Yesterday I talked in length about the DMPK and toxicology, and then I told you about that how the DMPK and toxicology playing a role. If DMPK toxicology you delay, you make a very potent compound, you made an IPR, you have set everything, biology looks good, you have a nanomolar compound, and then you give the compound to the DMPK to test the drug ADME property, all of the sudden the compound doesn't work, so you wasted around a 6 month or 8 months, your compound is a useless, you have to go back to the board, you have to make it, and to combat the activity which you are not getting in the ADME property, you can destroy your compound and then your tweaking can change and you will not get an activity also. So more important is early assessment of those compound as early as possible to have a feel of that compound is important, and if you get delay you are delaying the whole process or you have to go back to the board and synthesize a new compound. Need of further early collaboration, of course, we need an integration with the DMPK toxicology, risk detection and improves candidate. So that's important. Now I would like to tell you pitfalls in drug discovery, a small image I wanted, target identification, hit, lead, clinical, these are the stages which we have. The best example, somebody has told you that he is making, he has done a model on dementia. I am really intrigued to know about that model who was talking of, because we all in industry, CNS is something, a target, we all want to work on it, but the problem is we don't have a very good model. And see the data, high failure rate in CNS, over 90% of CNS drug fail because we don't have a मॉडल। जाहिर सी बात है इंसान की जो सोचने और समझने की क्षमता है वो जानवरों में नहीं है, वो रास्ता नेविगेट नहीं कर पाते हैं और उनका ब्रेन अलग तरह से काम करेगा, तो हम कैसे उस मॉडल को रेप्लिकेट करें, इसीलिए उसके छोटे छोटे हिस्से को रेप्लिकेट करने की कोशिश की गई है। एंड दे हैव डेवलप्ड अ मॉडल, कुछ स्विम मॉडल हैं, कुछ कैसे मॉडल्स हैं उनके पास, दे डू इट, बट नथिंग इज अराउंड 10 और 20% नियर टू द परफेक्शन, इसीलिए बहुत चैलेंजिंग, इसीलिए कोई भी CNS की दवा नहीं है ज़्यादा, जो कुछ है वो सुलाने की है, एंड ब्रेन वर्क्स इट्स ओन वे। तो दूसरा है फेज 3 फेलियर ओवर 30%, ट्रांसलेशनल गैप, एनिमल मॉडल, दिस इज इंपॉर्टेंट, whoever is a budding scientist, who are a biochemist, biologist, zoologist, who really want to work, I can tell you, focus of animal model. Do something new to the animal. People will take you like something. I paid to Tel Aviv University $30,000 for one model. And that was basically a PRMT CNS compound which we have given to them. That's one model, but was a niche area. If we would have developed, it would have taken 1 year to develop a method. But they have done in 1 week and sent. For me, the time is essence. So I every time say to the academia that you develop one assay. In industry, we don't want to develop assay. We want to do something, give you a compound without naming it, you give me a data, we will put it. But only thing is you should have a very strong data, you should have a very collaborative data with you. So people say then you have AI driven stratification, adaptive trial and organized. I will tell you some stories now, make you feel better, to have some interesting stories how it became. This is a story of Phyalurudine, and it's a very interesting story, you know. It was an antiviral drug that showed a promise in treating hepatitis B. However, during the clinical trial, it caused a fatal liver toxicity in several patients. The underlying cause was mitochondrial damage, which had been detected in preclinical studies, and tragic outcome highlighted the withdrawal. See, this was the drug, this was the antiviral. We could not catch in toxicology study. If they would have caught in the toxicology study, we would have got something different. They were withdrawal. Okay? So, important is this was something a pitfall. If we remember it, we would. So, what is the take home message? Take home message is need for the human relevant model. The case emphasizes the importance of developing predictive human relevant drug testing. So, what the message is for you guys? Think of it how you can do anything with a human which is more relevant you can produce in an animal or in vitro setup. That's the crux of the matter. I know it's not easy. People talk of AI and all, but AI will only be enriched if you give some data to them. So, you are the main scientist. When you sit on a lab, think of those models, how you replicate that system in an animal or in a petri dish. And that take home message for the people who really want to do a good research, think of it. Get your idea, write a grant to the people, and if it is relevant to the industry, industry will fund you some amount to start with the money. Then there is another story of TGN 1412, that was a cytokine storm which happened. This was tested in phase 1 trial to treat the autoimmune disease and leukemia, but triggered severe immune overreaction. That was one of the things. And what happened during the discovery or a development process, animal model failed to predict the human response. Immune response is still a challenge, although that's why when you do immune response study, we have to go, the second toxic species is every time is a monkey. Monkey is more relevant, they have more immunologically more relevant to human being. That's why we take that model. And because the study is so expensive, we don't do a very exhaustive study, and these are the things which happen. Checkpoint inhibitors are best example. You have seen Keytruda and many things you are talking of. They have been the lifeline, and now you are. So this was more important. So important was the changes in trial. The incident prompted the improvement in the clinical trial. Earlier they did it, and then the one patient died also in this trial. And that was one of the hallmark to that. There is another example of Vioxx that was it was a widely prescribed for pain and inflammation, and initially considered safer for GIT tract, better than NSAIDs. NSAIDs, you know, निमोसलाइड्स खाया होगा, नहीं खाया. नाइस टैबलेट किसी ने खाई होगी. इंडिया में तो चल रही है, बाहर बंद हो गई है, हैं? तो मत खाना कभी तुम लोग नाइस भी हैं. खैर लोग बेचते बहुत हैं, बिकती बहुत है. सर में दर्द हो फौरन नाइस दे देते हैं, हैं? तो कोशिश करो कि नाइस को ना ही लिया जाए तो अच्छा. नाइस इज़ नॉट नाइस. ओके. सो उसके बाद क्या हुआ कि दिस वाज बेसिकली इट्स ए COX-2 inhibitor. Sir might be knowing more, and when he went to that lab and all in those days, these were the thing which was growing like something, I know. And now people are not doing much work on it. Cardiovascular risk became apparent through the post marketing surveillance. पहले नहीं पकड़ पाए, डेवलपमेंट में नहीं पकड़ पाए, क्लिनिकल ट्रायल में भी पकड़ नहीं पाए. उसके बाद क्या हुआ, जब वो मार्केट में गई और बाहर जब गई तो पोस्ट लॉन्च के बाद मालूम हुआ कि वो जो है बहुत ज़्यादा जो है कार्डियोवास्कुलर रिस्क उसमें आया. अगर हम प्री क्लिनिकल टॉक्स में अगर हम अच्छा सीवी कर लेते, डॉग में सीवी किया जाता है, कार्डियोवास्कुलर डिजीज जितनी है वो डॉग में की जाती है और सिंगल डोज हम लोग देते हैं और उसके बाद रिपीट डोज की 5 डेज की स्टडी होती है, ज़्यादा देर नहीं. एक डोज देखते हैं, उसके बाद उसका ECG देखते हैं क्या है. 1 month 1 month 2 month 3 month 4 month 5 month 6 month 7 month 8 month 9 month 10 month 11 month 12 month 13 month 14 month 15 month 16 month 17 month 18 month 19 month 20 month 21 month 22 month 23 month 24 month 25 month 26 month 27 month 28 month 29 month 30 month 31 month 32 month 33 month 34 month 35 month 36 month 37 month 38 month 39 month 40 month 41 month 42 month 43 month 44 month 45 month 46 month 47 month 48 month 49 month 50 month 51 month 52 month 53 month 54 month 55 month 56 month 57 month 58 month 59 month 60 month 61 month 62 month 63 month 64 month 65 month 66 month 67 month 68 month 69 month 70 month 71 month 72 month 73 month 74 month 75 month आज नहीं है क्या? ये एक और एग्जाम्पल है BIA 102474 and Sam might be knowing it because it's more from his own company earlier. तो उन्होंने फा पे काम कर रहे थे, फा एंजाइम पे बहुत Oh wow, yeah, I can tell you. No. Yeah. Actually, what happened, this also comes from something known as a confidentiality thing. And there is a lot of work goes on. Like in our company, we give JBI number, JBI 278, 778. So what happened when a program starts, there is a numbering, there is a company name which goes on, and then you have a how much the compound is made. Total, when you submit a compound, there is a known as a compound submission form or a compound when you start working on it, there's a lot of compound you put it. So you start making a compound. When a project start, because this is every places has a different way, they do project wise. Somebody knows how much the discovery project has already been moved, or in a particular project, in our company, in a particular project, how much compound we have made and where we reach it. We start with a similar number. It's sometimes chosen also. Some people start with the 700 and they do 700, 1, 702, 778. Means this 778 means they must have put 7,000 compound or 80 compound or 90th compound has made. So it has a confidentiality, but the first number usually, like this BIA is basically from Bayer number, if I remember rightly. They also write BAY also, their number, and he Eli Lilly put actually they put LI number. LI they put it, many people different name. Pfizer every time put PF number. So if you see and put a number, they will say PF, and that will be the number which they will be writing. You can understand from where the compound came. But the numbering is basically how much the compound has been synthesized. That has not much of importance. People have a different strategy so far, but this is how it is done. Anything I want to add, sir?
Thank you.
That's the only thing. There is no norm like this. It's a code, and we don't want that to spill it, okay? If we give some name, then it will be issue sometime. Okay, 2471 neurotoxicity. This was one of the drug which was a Fa enzyme. Fa is, you know well, Fa is a target which was explored way back, you know. Yeah, Fa was basically a fatty acid amide hydrolysis. पेन मैनेजमेंट के लिए यूज़ करते थे. फेज़ 1 में फ्रांस में इसकी ट्रायल जब हो रही थी तो एक पेशेंट मर गया ट्रायल में, और ये किसी भी ऑर्गेनाइजेशन के लिए एक पहाड़ उसके ऊपर फेंक देना. इसीलिए मेरी जॉब कभी कभी बहुत इम्पॉर्टेंट हो जाती है, क्योंकि मुझे जो है, मैं हूँ FIH डिसाइड करता हूँ अपने आप. FIH का मतलब फर्स्ट इन ह्यूमन डोज है, वो एनिमल डाटा से लेके हमको बताना. अब वो 10 टाइम लोअर होती है जो एक्सट्रीम टॉक्सिसिटी हो सकती है, मगर ह्यूमन एक अनप्रिडिक्टेबल चीज़ है. यू ऑल नो वेल, आई टोल्ड यू दैट विद द वार्म ऑफ ए हैंड, चेंज ऑफ ए pH, and any shake of something, if something change the structure, why don't with the building block of something will not be a stable or unstable people. That was Isaac Asimov's story. So what I want to tell you guys is अनप्रिडिक्टेबल। होता क्या है, हम मॉडलिंग करते हैं, मैं मॉडलिंग भी करता हूँ, सिमुलेशन जिसको कहते हैं कि एनिमल में इतना आया है, ह्यूमन में इतना आएगा, क्योंकि इसके लीवर का रन इतना है तो मेटाबॉलिज्म इतना कम हो जाए, मगर ये होता नहीं है। आज की डेट में मैं अपना डाटा भी बता नहीं सकता हूँ, बट देयर इज ए ह्यूज थिंग, आई एम सीइंग व्हाट आई वाज प्रिडिक्टिंग, इट इज आई अंडर प्रिडिक्टेड समथिंग which I was thinking that 60 or 80 we will get, we are getting activity in just 40 also. So important is the exposure and many things. So why I am saying it, तो जब फेज 1 में जब वो पेशेंट की डेथ हो गई तो उन्होंने कहा कि क्या वजह हो सकती है, और ये पाया क्या गया, तो एक ऑलरेडी उसमें ड्रग था, तो उन्होंने कहा कि क्या हो सकता है, ये क्या है इसको, ओके न्यूरो, एंड बाद में क्या हुआ कि ये क्या हो सकता है, हुआ क्या कि लिपिड मेटाबॉलिज्म को ये अफेक्ट करता है, तो ब्रेन में जाके ब्रेन की जो लिपिड थी उसको उसने डिसॉल्व कर दिया, और वो नहीं होने की वजह से न्यूरोटॉक्सिसिटी आई और उसका फिर विड्रॉल आया। तो हुआ क्या कि अगर आपके पास अच्छा मॉडल होता, प्री क्लिनिकल में न्यूरो का मॉडल अच्छा होता, आप उसी वक्त टॉक्सिसिटी उसकी चेक कर लेते और आपको ये इशू नहीं होता। तो आप क्या फ़ायदा होता, एक तो इतना पैसा बच जाता, दूसरा आपका वक्त बच जाता, और तीसरा वक्त के साथ लोगों की इमोशंस जुड़े होते हैं वो बच जाते, और सबसे इंपॉर्टेंट है आप कुछ और नया बना चुके होते। अब आप इतने साल आने के बाद फिर वापस बोर्ड पे जाएंगे, फिर एक नया काम करेंगे, और देयर इज अ लॉट ऑफ रिसोर्स प्रॉब्लम। सो the take home message so far I have given is, we don't have a good model, we don't have a preclinical model which is good enough to predict in human. This is a scope for academia, this is the biggest scope for academia. I am telling you, people are talking lot of drug discovery, scientists, they becoming a drug, they want to launch the compound, they have a patent, they say we can do it. It's still very nascent. I want you guys to sit on a lab, make a small models, that's all, and you will succeed like something. People are ready to pay you $10,000 without asking question if you have a relevant model with some proof of concept if you have. Another is a methadone story, we all know well, maybe it was a big story which was happened, it was a calcium channel blocker. And then there's a dangerous interaction, DDI issue was there. DDI, I explained yesterday how the DDI works. One drug works, another drug if you give along with that, it doesn't work due to CYP and all those metabolism, transporters and everything. So that was one of the reasons. This was a very wonderful story where FDA was caught red-handed, okay. And this was a story where the controversial approval happened, and this compound was cognitive improvement for the Alzheimer disease. उसी टाइम 10 लोग बैठे थे सेफ्टी की एडवाइजरी कमेटी में, और 10 लोगों ने 10-0 से इसका अगेंस्ट किया था, फिर भी इसको अप्रूव किया गया, और अप्रूव करने के बाद फिर ये इफेक्ट नहीं मिला, और उसके बाद जब नहीं मिला तो 221 में अप्रूव किया गया, और उसकी सेफ्टी और लैक ऑफ एफिकेसी की वजह से 2024 में इसको विड्रॉ भी कर लिया गया। तो यहां पे क्या है, रेगुलेटरी इशू है, ये पिटफॉल्स है, ये सब वजूहात हैं जिनकी वजह से हम ड्रग डिस्कवरी को हमने इतना डेकोरेटिव तो बनाया कि बहुत अच्छा है, इंटरेस्टिंग है, ये पैसा कमाने का है, ये नहीं, चैलेंजेस बहुत हैं, और जब गिरता है तो वो उसको ज़मीन पे गिरता है, फिर तबाह बर्बाद हो जाती है, बड़ी बड़ी छोटी कंपनियां तो गायब हो जाती हैं बिल्कुल सतह से। तो इसलिए ये बहुत इंपॉर्टेंट है, फूक फूक के कदम, हर डिसीजन आपका बहुत एक कंपाउंड हम जो एक और मैसेज है बडिंग साइंटिस्ट के लिए, स्टूडेंट के लिए, ये आप इतना पढ़ा नहीं पढ़ा, रख लो ना रख लो, मुझे कोई फ़र्क नहीं है, आपको भी ज़रूरत नहीं है, आपको ज़िंदगी में काम आएगा, कभी आप सोचो। एक चीज़ याद रखना, बी ऑनेस्ट विद योर रिसर्च। अगर आपने एक बफर भी बनाया है और PS 7.4 अगर बनाया है तो 7.4 लिखो, 7.2 बनाया 7.2 लिखो। ये नहीं है कि हमने बनाया और हम जल्दबाजी में 7.4 लिख के चल दिया। कुछ भी, कोई भी काम आप करो, अगर ऑनेस्ट रहो, नहीं दिल चाह रहा मत करो। ये आपकी रिसर्च आज आपको गलत नहीं बताएगी, आगे सुपरवाइजर देख के हाँ बहुत अच्छा डाटा है, बहुत अच्छा पीक आ गया, बहुत वंडरफुल है, आपका कंपाउंड अच्छा नहीं। आप ऑनेस्ट हो जाओ, वो आपके ज़मीर को जवाब देना चाहिए कि हमने, ये बहुत फ़ायदा करेगा अपनी ज़िंदगी में। हम जानते हैं कि लोगों ने मॉलिक्यूल को देखा, उसको उठा के कह दिया स्टेबल है, दे दिया, देने के बाद उसमें तो एरर था, उसने बताया नहीं, इस कंपाउंड को उधर डाल दिया, इधर को उधर डाल दिया, जो भी किया, बाद में पीके हुआ, पीके के बाद भी डाटा समझ में नहीं आया। पीके $2000 का एक पीके होता है, अगर वो वहाँ पे एडमी में रोक लेता, पीके में नहीं जाता, हम $2000 बचाते। कहने का मकसद है कि बहुत ऑनेस्ट हो जाओ रिसर्च पे, रिसर्च से ज़्यादा ऑनेस्टी बना के लिखना कोई ज़रूरी नहीं है। छोटा काम करो, सही काम करो, गलत आया डाटा वो छापो, गलत डाटा ने बहुत रिसर्च बढ़ाई आगे। प्लीज डोंट डू दैट। आई डोंट वांट टू यूज अ वर्ड प्लैगरिज्म, आई डोंट वांट टू स्पीक इंग्लिश। ये हमारा ईमान है, वी हैव टू बी ऑनेस्ट विद द रिसर्च। जो भी काम करो ऑनेस्ट, किसी को खुश मत करो। छोटा भी बफर बनाओ तो बफर ऑनेस्टली बनाओ बस, ठीक है। आई थिंक दिस इज द बेसिक मैसेज विच यू शुड कैरी विद यू गाइस। Okay, I will give you some of the quickly. This pitfall is the poor target validation. I already give you some example where you can do multi-omic CRISPR. You all know well. If somebody knows CRISPR or not, I don't know. But this is something, a technology we should know. Inadequate translational model, homogenized model, and organoid. Admit issue, early in silico and in vitro. These are the strategies people can follow. But it should be, there should be a validated method when you are talking of any of these things with respect to in silico. Anything biomarker misuse, that's the biggest problem which is having. The two science which I want to give you a message in AMU that you should focus. One is the biomarker and other is the model. These two things if you can do, you can go somewhere else. You don't have to think backside where you are. Data in silos, don't put your data in silo. Discuss among your friends and talking to you and see यार मेरे को ये हुआ, मैंने ये जेल रन किया, इसमें ये नहीं आया, क्या हो गया, कहीं मैंने ज़्यादा कंसंट्रेशन का तो कुछ नहीं मिला दिया था, ये प्रोटीन इतनी दूर गई, ये नहीं गई, ये हम कैसे इसको निकाल सकते हैं एक्सट्रैक्ट। आपस में डिस्कस करो, कॉफ़ी पे डिस्कस करना ज़्यादा इंपॉर्टेंट है एंड यू विल सक्सीड ऑन इट। दैट इज़ द मैसेज विच आई लर्न इन मैनी ईयर्स ऑफ माई डूइंग रिसर्च। Innovative approaches to the overcome, I already told you, that is just a thing which we have already done. Why integration is matter? ये क्यों इंटीग्रेशन चाहिए? इंटीग्रेशन मैं बार बार बोल रहा, देखो इसका टॉपिक भी है इंटरडिसिप्लिनरी। हम लोग जो ड्रग डिस्कवरी कर रहे हैं वो एक आदमी का काम है ही नहीं, ये सबका काम है। कोई कहे कि मैं करता हूं, हो सकता है एक लीड करता है, एक बीच में खड़ा हुआ है, वो सबको देख रहा है, वो विजनरी है होगा, मगर वो एक आदमी ये काम नहीं कर सकता। तो इंटीग्रेशन इंपॉर्टेंट है, दैट इज व्हाट आई एम सेइंग, इट विल इंप्रूव द कैंडिडेट सिलेक्शन। जाहिर सी बात। और एक और चीज है, व्हेन यू टॉक योर बॉसेस और यू टॉक ऑफ योर सुपरवाइजर, डोंट surrender yourself with your thoughts. I'm not saying you start giving backside to him, whatever, but don't surrender with your thought. Novel ideas are everything. So similarly, my boss may be one of the project leader, he may say, Zain इसको कर दे और ये हो गया, ठीक ठाक है, कंपाउंड बहुत अच्छा। हम कहेंगे नहीं, कंपाउंड अच्छा नहीं है, ये मेरी नज़र में कूड़ा है, इसको टेस्ट करो, नहीं तो फेंक दो इसको। तो क्या होता है कि उससे फ़ायदा क्या है कि आपका कंपाउंड रोक रहे हो, उससे आप आगे की रिसोर्स को कहीं और इस्तेमाल कर सकते हो, ठीक है। तो देयर इज अ साइंस बिहाइंड इट। अर्ली इंटीग्रेशन एलिमिनेट्स वीक ड्रग कैंडिडेट बिफोर मेजर रिस्क, एनहांस प्रेडिक्टिव टॉक्सिकोलॉजी, predictive toxicology, that is a failure for that, and boosted the translational relevance, that what I was talking about. Aligning the preclinical finding, that's more important, that's more of a translational science. When you do a preclinical finding, उसको अगर clinical से अगर relevancy आपको मिल जाएगी, तो ये सबसे बड़ी चीज़ है एक drug discovery के success होने का। I believe in this concept more than anything else, concept of fail fast and fail smart. See, nobody want to fail. Chemist, every time we have a fight. I belong to a department where we are said killer. The chemist has a baby, you know, वो उसको पालता है, उसको बड़ा करता है, पैदा करता है, जैसे ही बड़ा आता होता है हम लोग कहते हैं ये बच्चा नालायक है, इसको आप हटा दो, ये बेकार का आदमी है, इसको फेंकिए, ये किसी काम आगे जाके तुम्ही को नुकसान पहुंचाएगा। मगर वो बेबी को कैसे कोई बच्चे को अपने छोड़ता है? इसीलिए हम लोग बोर्ड रूम में और कहीं भी डिस्कशन में लड़ाई बहुत होती है, समझो मारा पीटी तक नौबत आ जाती है कि ना आप इसको फेंकना चाह रहे हो, आपको दूसरा नहीं, इसको ऐसे कर लो, ऐसे कर लो। दिस आर द साइंस विच वी हैव डिस्कशन, एंड ऑफ द डे वी ऑल एग्री टू द डिसएग्रीमेंट। ओके, तो इसमें अर्ली इंटीग्रेशन आई ऑलरेडी टोल्ड, बूस्टर ट्रांसलेशन, फेल फास्ट में वही चीज़ें हैं जो हमने पहले बताया। चैलेंजेस टू द इंटीग्रेशन, ये सबसे बड़ा चैलेंज है इन एनी ऑर्गेनाइजेशन। क्या होता है कि आप डीएमपी के टॉक्सिकोलॉजी को सेवरल चैलेंजेस की, ऑर्गेनाइजेशन इन साइलॉस में काम करते हो, फ्रेगमेंटेड डाटा सिस्टम होता है, हर आदमी अपना अपना डाटा उठा लेता है। अब जैसे डीएमपी का कभी डाटा एक उठाया उसने, एक्टिविटी सिर्फ देख ले, दूसरा चीज़ नहीं देखे, कोई फ़ायदा नहीं। इसीलिए जो प्रोजेक्ट लीडर बनता है ना, उसके अंडरस्टैंडिंग सबके लिए। तो हम लोग जब स्लाइड बनाते हैं तो उसमें हम एक कंपाउंड का स्ट्रक्चर डालते हैं, हम उसकी एक्टिविटी डालते हैं, एक्टिविटी के साथ उसकी नॉर्मल एक्टिविटी भी डालते हैं, एंजाइम एक्टिविटी भी डालते हैं, सेलुलर एक्टिविटी भी डालते हैं, फिजिको केमिकल प्रॉपर्टी भी डालते हैं, जैसे सॉलिबिलिटी है, पीके है, मॉलिकुलर वेट कितना है, रूल ऑफ 5 यस्टरडे, लिविंस्की रूल ऑफ 5 यस्टरडे, वो कहाँ फिट होता है। तो वो करने के बाद फिर हम लोग एडमी प्रॉपर्टी पूरी डालते हैं, फिर पीके डालते हैं, फिर एफिकेसी की भी एक स्लाइड होती है, एफिकेसी नहीं है तो पीडी मार्कर होता है, और उसके बाद एक स्लाइड में सब होता है। तो हर आदमी की नज़र में पूरा कुछ हो, प्रोटीन बाइंडिंग भी है। अब एक कंपाउंड है 90% प्रोटीन बाइंड है, एक है 99%, और सब कुछ सेम है, तो मैं 90 वाले को ले लूँगा, 99 वाले को फेंक दूंगा, डीप्रोटाइज कर लूँगा, ठीक है। फिर आगे जाके देखेंगे अगर फेल होता है तो फिर इसको बढ़ाएंगे। तो कहने का मकसद ये कि डाटा को हॉलिस्टिक देखो, डाटा साइलॉस में नहीं देखा जाता है। आप एक डाटा को ना देखो, सबको देखो। सॉल्यूशन दैट वर्क, ऑफ कोर्स जॉइंट प्रोजेक्ट टीम, ये हम लोग की हमेशा होती है। हमारी जब हम लोग प्रोजेक्ट टीम मीटिंग करते हैं तो एक केमिस्ट होता है, एक प्रोजेक्ट लीडर होता है, कभी वो बायोलॉजिस्ट होता है, कभी वो केमिस्ट होता है, एक कोर बायोलॉजिस्ट होता है, एक डीएमपी के एडमी टॉक्स का आदमी होता है, एक आदमी जो उसमें होगा वो कॉम्पिटिशनल का होता है, अर्ली ऑन, बाद में नहीं रहता है वो। तो उसके बाद जैसे जैसे जिस स्टेज पे होते हैं वैसे तो एक पूरी टीम बैठती, and there is 1 hour, there is a huge discussion and all those things which goes in that place. Shared data dashboard, that's the way we do early risk assessment, और वहीं से रिस्क भी असेस हो जाता है कि अगर इसको ले जाएंगे तो ये इशू है। और एक सबसे इंपॉर्टेंट है कि जो हम लोग बार बार देखते रहते हैं, एक टीम होती है जिसका काम है कॉम्पिटिटिव इंटेलिजेंस देखने का। आज मैं यहाँ पहुँचा हूँ, दूसरा कंपाउंड कहाँ है, मान लो फेज़ 2 में पहुँच गया है, तब हम उसको एसेस करते हैं, ये निकल जाएगा कंपाउंड, अगर हमको पहुँचने में 10 साल, अभी 8 साल लगेगा वहाँ पे, तो हम इससे कुछ पैसा बना पाएंगे कि नहीं, या कोई फ़ायदे की बात नहीं। तो हम उस वजह से प्रोजेक्ट को रोक भी देते हैं, उसको कहते हैं स्ट्रैटेजिक डिसीजन से हमने उसको होल्ड कर दिया, ठीक है। या उसका फेलियर आ गया फेज़ 1 में, तो हमने कहा यार वो तो फेल हो गया, टॉक्स उसमें दिख रहा है, और अब फिर देखा जाता है क्यों फेलियर हुआ है। फेलियर टारगेट की वजह से है या फेलियर उसके कंपाउंड की वजह से। अगर टारगेट की वजह से है तो फिर तो नहीं यूज़ करते हैं, तो उसको रोकना ही पड़ता है। This is the next gen discovery vision for the future. AI, ML, still we have to write. I am looking for some of the more data to be more authoritative in saying this thing. System biology model, this is what I am emphasizing you guys to have some good model which can help you. Personalized pharmacokinetics, that is something important. That somebody asked me a question yesterday कि इतना कुछ होगा तो एक आदमी के लिए ड्रग क्यों डेवलप करेगा। वो इसलिए नहीं करेगा कि एक आदमी के लिए, उस ड्रग को ट्वीक करेगा। सबसे अच्छा तरीका है कि अगर उसका पीके पता चल जाए कि उस कंपाउंड का पीके इस आदमी में कैसा है, तो उससे हम सिमुलेट कर लेंगे कितने पे इसको एफिकेसी। किसी ने 1 ग्राम देने पे 3000 उसका एक्सपोजर है, किसी को 1 ग्राम देने में सिर्फ जो है 300 एक्सपोजर है, तो 300 वाले को हमको 10 ग्राम देना पड़ेगा, और उसको तो इसको कहते हैं पर्सनलाइज फार्माकोकाइनेटिक स्टडी, ठीक है। एंड देन यू हैव अ पैराडाइम शिफ्ट इन ड्रग डेवलपमेंट, दैट इज हाउ वी हैव टू डू। Important is a global high. I was telling a story when I was talking to Eli Lilly guys and said, why don't you do one thing, put a platform, put your failures in a data. I think the discovery will be more cheaper and more faster and everybody can succeed. You can come with a common platform, but from this data, if somebody develop, they can share the revenue. He said it's not so easy to work on it, and that was one of the answer, one-liner answer he gave. I know that was just a question, but still. So key takeaway, importance of integration, DMPK and toxic reduces risk and this. We can add some other department. Summary of the tools and overflows, that what we all have talked length in our collaboration is the key. This is most important. Cross-disciplinary teamwork is essential to successfully integrate the latter stage of drug discovery, and improved outcome and the cost reduction can only happen if we integrate smartly and closely. The take home message is, let's build bridges, not silos. Is it okay? And that doesn't goes in industry, in our DMPK or in our drug discovery and development. This also goes in your labs. So for you, you have to take for yourself, sit together, discuss. Don't keep everything close to your chest. Discuss. Things should be kept on a close to chest, but science, discuss, you will succeed. I think that's all I have. If you have any questions, please shoot. Yep. Mm-hmm.
So my first question is, like we are having some toxic drugs now which failed during the clinical studies in human trials, and they are having neurotoxicity or cytotoxicity or whatever it is. So is it possible that as we have spent billions of dollars in the production of those drugs, is it possible now that we can reuse, or we can use a certain different application of these drugs for treating cancers? As I have came across, there was a drug, thalidomide, which leads to the fetal abnormalities. So at that time, that drug was banned, and now people are using that drug for treating cancer. Is it possible now, because we are using those drugs now that has already been prepared?
Good question. So you want to take it because it's a thalidomide for you, but
I have to come here.
yeah.
This is a very, very fascinating story. Thalidomide was developed for leprosy. Okay, and then after some time, this is really a very, very good point you raised. Thalidomide was developed for leprosy. Then there was they started using it, and they found that there is a fetal issue, means the children were born without hand and legs. Okay, and then it was banned. Now thalidomide come back because cancer patient, what we say, either you kill the patient or you kill the drug, right? So they said, okay, we will not kill the patient. Okay, we'll use the drug. So that started working in especially MLL or myeloid leukemia. Now thalidomide, you might not have seen that this is the most important part of the PROTAC or protein degradation part, because this is the one which goes and binds into E ligase enzyme. Okay, and we call it CRBN. That is the thalidomide or its analog. There are two we use. There are many more. So that was so how you said that okay, can the drug be used for which is very toxic and be used for different things? This is the one which is very important, has become very important, nothing to do with toxicity, because if you use that alone, it is toxic. If you connect with something, it is not toxic. I gave an example of colchicine yesterday. Very toxic, 0.0007 nanomolar. If you give, it is toxic even at that level. But when you connect to something, it becomes bitter. So yes, the drugs which are neurotoxic or cytotoxic can be used for some disease, but you have to do a lot of work on that. Okay?
Yeah, that's what yesterday so I was telling it. So I asked him to tell you a story. But one more thing I can add you when you do a toxic study.
There
So
are many drugs like
yeah,
that we don't have time to
and
tell you.
thalidomide, if you handle thalidomide, you can't stay in a lab for a long time. So that's having such a smell, you know. I worked with a guy who really worked on that thalidomide. He was a Pfizer guy. He came and taught me some assay with something, and he was making. I want to tell you a story with what we do. When we do a tox study or a human trial, there is a question that whether it should be given to pregnant women, where it should be given to females, whether it should be given to the. So there you have an option to tell no, we will not treat it. There we will. So these are also the options if you understand them. And the second most important is more important. You gave an example, thalidomide. We gave a story, but in a generalized form, the story is simple. You have to understand the tox. Is it a tox coming from a target, or is it a tox coming from a molecule? Fine. If it is target for the target, fine, that target may be useless. If molecule is not there, you can check with the other target, do a quick study which is known as a bridging study, and then you can move forward. There are many drugs which have been, people are doing a repurposing. That's the reason, even with a successful and an unsuccessful drug.
So for example, in case of calcium-MIC, it was gone after 7 years on the market, but that was a mistake by a physician. There is no physician here, right? No doctor, otherwise they will they will tell me something. So it was a mistake because they used to give a very large amount of drug one time, once a day, injection. Now somebody said, no, this drug is very, very effective, although it is very toxic. Okay, why not we give at 6-hour interval? And they started giving at 6-hour interval. That drug is still in market and making billions of dollars. So, you know, cancer drug, very toxic drug, cancer drug, can be modulated by little bit of, thinking into a very nice product, and it is treating a lot of patients. The survival rate using calcium is very high. Yeah, that is another one. Yeah, it was used long time ago in World War I in for syphilis, and World War II also. Now arsenic is being used for some other purpose also. Yeah.
Anything else you have? Yeah, go ahead.
Sir, during clinical trial, how you handle the volunteers, sir, in phase 1 or phase 2?
Very tough job. Very, very tough job.
How that I don't know.
See,
how, sir,
when we do a research in our clinical trial in my company, we do with the patient actually. But patient also, it's tough, you know. They are already emotionally stressed. They suffer a lot. There is a lot of people who are giving. I do with lung cancer. So people are terminal type. They are told that you will alive for 15 days, you're 10 days or something, and they come to the trial because they don't have any option outside. Then they get a lot of suggestion, falana baba hai, unke paas chale jao, falani cheez uske. It's tough. But the only thing is the doctor compliance or doctor confidence bring them to inside, and we try to save. And many of the people, one of the patient, I will tell you a story from my drug, and it's in US. There is a guy, startup is our physician. There was a guy who came, he was a 54-year male, and he came and he was said that you start preparing yourself, you have another 1 month. Okay, and then he came to us, and we startup asked him that this is one of the trial going on, and that is more of STK11 story I'm telling about. So we told that okay, you can take it and just you can move forward already. All of a sudden, that drug, that patient came to it, and today it's around 2 years, 4 months. He is still on the drug. Now he is going on vacation. He, as a kid, he dropped his son day out and day out. He's normal. Only thing he tells that I pray for you guys and you keep on giving us compound. Okay, so this is how it is. This is, we made a difference at least for one. There will be lot to come. Yeah.
Because sir, for some disease you need 10 to 200 patients having the same disease, and we are checking for a drug. So we need patient which is having this disease with which we are working, or you are able to
Yeah,
check.
788, 800, 900, 1100 type of a cohort studies are done, you know. So that's a take 10 years, 11 years, you know. Hmm? Okay.
The next session was supposed to be interactive. We all three were supposed to, but I will start.
फिर ये देखना है कि उसकी सेफ्टी भी है कि नहीं, ऐसा तो नहीं है। इसको देखें, उसका फेज 1 है, लोगों पे होता है इस तरह से स्टेप, ठीक है ना। ये प्रोग्राम
तो सही कह रही है, स्टडीज पे भी ध्यान दें।
आप एक पेज निकालो, उस पे लिखो
So did you decide upon the indication that we can have? So just one cancer, nobody can get it, cardiovascular so this is about how would you go about finding that write it down, make some figures, because we have to present each one of you, not every one of you, at least 2 people can come over.
जिसको हम लोग कहते हैं टॉक्स। अब टॉक्स करने के बाद आपने देखा कि आपका क्या टॉक्स आता है। टॉक्स हमने किया 2
क्या डिस्कस
अब आपको स्ट्रेटेजाइज करना है, एनडी बनाए। अब एनडी के लिए इसको कहते हैं, अब पेशेंट जा रहा है फर्स्ट एंड ह्यूमन, तो यहाँ सबसे पहला काम होगा फर्स्ट एंड ह्यूमन डोज इसके बाद आप पेशेंट देखने की भाई जाहिर सी बात है, जब आपको डोज पड़ेगा पेशेंट कौन से डिजीज़ पता है, कैंसर है मान लो, कौन सा मान लो लंग कैंसर। अब लंग कैंसर के लिए आप साइट देखने पड़ेंगे, डॉक्टर से मिलना पड़ेगा, उसकी एक फेज़ 1 वहाँ पे आपको सेफ्टी क्या मिलेगी कि ड्रग जितना देने के बाद पता चला कि 2 mg से आपने शुरू किया और 10 mg जैसे ही पहुँचे पेशेंट जो प्रॉब्लम देखने तो 10 mg से ऊपर हम नहीं जा सकते। अब आपका बेस्ट डोज 10 mg था, 10 mg पे आपने फेज़ 2 किया, क्या किया। अब यहाँ एफिकेसी देखेंगे, क्या देखेंगे, बस इतना एक प्लान बना बैठा कि आप क्या करोगे, किसके जाओगे, मेडिकल में जाओगे, वहाँ डॉक्टर से मिलोगे।
कॉन्फिडेंशियलिटी का ख्याल रखिएगा, आइडिया चोरी ना हो जाए।
इसमें आप लोग एक चीज़ ये भी देखिएगा कि कौन कौन फंक्शंस हैं, कौन कौन सा
डिपार्टमेंट में बात करी थी तो डिपार्टमेंट वाले एग्री हैं इस पर, लेकिन इसका मैं पूरा प्रपोजल बताऊँगा। इस पर फिर हम
इसके अंडर में अंडर में टीचर के बारे में रेगुलेटरी एजेंसी को यहाँ से देखेगी
और एमी की बात
दे देगा।
डिपार्टमेंट के लोग
ये जवाब नहीं हो सकते हैं, डिपार्टमेंट के लोग ऐसे दे रहे चलो उसको हम बात करेंगे पहले जैसे तो हम नहीं प्लान कि हम नहीं नॉन रेगुलेटरी स्टफ उसके बाद एजेंसी काम करती है, कमेटी काम वो एथिकल कमेटी से वहाँ पास करने के अंदर आपको आगे सबमिशन कौन देगा, आगे नेक्स्ट लेवल पे सबमिशन कौन देगा, आप एप्लीकेशन कर सकते हो, उनका डाटा क्या निकालोगे।
वो हम
क्या
जैसे कि एस्मा में
साइटोकाइनेसिस देते हैं?
साइटोकिन्स मार्कर्स होते हैं और
तो विद इन फर्स्ट में भी अगर
अगर तो वो अगर नॉर्मल इस टाइप से रहता है बाद में
क्योंकि मोस्ट ऑफ द ड्रग्स
या फिर मैं भी स्टडी कर रही थी इसको टॉक्सिसिटी के लिए है कि
करना पड़ेगा ना। आप इसको बनाओ जल्दी सब लोग मिलके एक प्लान बना लो कि एक प्रपोजल है कि हमारे पास उन्होंने ये मॉलिक्यूल दे दिया तो अब देने के बाद अब आप कैसे गए कि ये एक पूरा एक रन करके बनाओ, सब लोग डिस्कस करो कि हम यहाँ करेंगे फिर यहाँ डीएमपी के घूम जाओ ना सब लोग यार, तुम लोग घूम जाओ सही से बैठ के क्या है अपने दिमाग में ये सब बताओ कि जैसे हुआ इन्होंने दिया, अब हम क्या क्या करेंगे, हम कहाँ जाएंगे, कहाँ टॉक्सिक स्टडी करेंगे, फिर किस जैसे करेंगे, टॉक्सिक स्टडी करने के बाद फिर हम किसी आइडेंटिटी करेंगे और फिर हम किसी रेगुलेटरी को सबमिट करेंगे, फिर वो परमिशन मांगेंगे, फिर तुम्हारा काम में तुम्हें सबसे पहले एडमिट करना है, एडमिट नहीं करोगे तो कोई पूछेगा नहीं, टॉक्स डाटा कहाँ से लाओगे?
इसमें टॉक्सिकोलॉजी टॉक्सिक इसमें वो बताता है। हाँ, इसमें वो है ना बेसिकली इसमें पैरामीटर होते हैं टॉक्सिकोलॉजी में IC50 और GC50 IC50 होता है इनहिबिटरी कंसंट्रेशन है 50% कंसंट्रेशन और GC50 होता है डेथ कंसंट्रेशन है 50%। तो
You can take the help of AI and everything, but try to apply what you have learned so far.
इससे हमें पता चलता है कि कितना कंसंट्रेशन है, यानी कोई जैसे सर ने बताया MT 10 MT 5 रीजन को पता लेना है तो फिर हमें यहाँ पता चलता है कि इसमें ड्यूटी इफेक्टिव है। दूसरा मैं उस पर चेक करा रोटेशन की ट्रेंड को 30 लेकिन ह्यूमन में 30 एम की नहीं चाहिए, मतलब भी पेश होने को ट्रायल ये तो 30 एम की तो दिखता है, इसके ऊपर तो लिखा है गोल्डेन
जब तक
लेकिन वो स्विच करना है इसमें जाने के लिए
प्रोटीन के बारे में
एनिमल
प्रोटीन इज वेरी तो मॉलिक्यूलर क्राउड जब हमारे सेल इंटरैक्शन तो जब हमने कंट्रोल कंडीशन लैब में टेस्ट तो बाइंडिंग हो रहा है, लेकिन मॉलिक्यूलर क्राउड में जब वो इंटर एक सेलुलर एनवायरनमेंट में जा रही है तो उन चीज़ों को क्या
इसी वजह से हम स्टेप बाय स्टेप इन सिलिको इन प्रॉपर कंडीशन आप जैसा चाहेंगे सब कुछ वैसे कर उसके बाद चीज़ें हम डेवलप कर सकते हैं
वो बढ़ जाएगी, पहले मान लीजिए 50 या वो कितने हो जाएंगे 100 का मार्कर 50 हो गया, फिर उस पर हम चेक करेंगे। अब उस पर भी कुछ आया कि नहीं 10 तो ज़्यादा हो रहा है तो उसमें भी कुछ डिपेंड डाउन किया 10 इफेक्टिव नहीं है तो बढ़ा भी सकते अब इसमें ये कि इसमें कुछ सारी चीज़ें जो मैं भी सर से डिस्कस करता हूँ, जैसे ड्रग का ट्रायल थोड़ा सा मुश्किल है, ड्रग जो कि वो केमिकली नेचुरल भी थोड़ी सी वो नेचुरल प्रोडक्ट्स हैं तो प्लांट के तो प्लांट बेस्ड जो कंपाउंड्स हैं उनको जाना ईजी है क्योंकि हमारा ADMT सब होता है विदाउट ADMT डोंट ठीक है। तो ADMT बहुत इंपॉर्टेंट है, ADMT मींस एब्जॉर्प्शन डाइजेशन मेटाबॉलिज्म एक्सक्रीशन एंड टॉक्सिकोलॉजी, उसको कहते हैं शॉर्ट एडमिट। तो ये सब चीज़ें होनी चाहिए, एब्जॉर्प्शन कैसा है, मेटाबॉलिज्म कैसा है, तो जो हर्बल मेडिसिन हैं, जो हर्बल ड्रग्स हैं, उनकी रेडमिट प्रोफाइल अच्छी होती है, क्योंकि नॉर्मली जो हम यूज़ करते हैं ना उसको, मान लीजिए कोई कलौंजी का तेल है, ठीक है, ऑनियन का तेल है, उसका तेल है, उसका तेल है, अब वो ऑयल क्या है, कोल्ड प्रेस है,
जब आप इसके बारे में भाई काम कर रहे हैं बता सकते हैं इसमें कार्डियोवैस्कुलर
तो ये रेडमिट प्रोफाइल टॉक्सिक इफेक्ट क्या है, लेकिन अगर आप उसको e-commerce दे रहे हैं तो गुड, बट
मेन हार्ट के लिए होता है।
तो इनसे में मुझे लगता है इन विट्रो
कितना यूज़ करो,
तो क्योंकि वो हार्ट से मायोप्लास पे ही टेस्ट करें तो वो कार्डियक मायोप्लास्ट क्योंकि वो हार्ट सेल्स ही है तो उनकी क्या कहते हैं अगर हम लोग फिर फर्दर टेस्ट तो उससे हमें पता चलेगा कि हार्ट सेल्स पर क्या असर पड़ रहा है क्योंकि हार्ट सेल्स H9C
Instead of
हमें ये दिखा के जो भी
लेकिन मतलब
अगर आप याद
मतलब हम जो भी सेल लाइन लेंगे तो कुछ तो रैशनल हमारे सेल लाइन उसको तो मुझे लगता है अगर हम मल्टीपल सेल लाइन कॉन्सेप्ट पे भी करते हैं तो एक हमारा सोचने में ये है कि हम लोग एक सेल लाइन ये भी रखें जो डायरेक्ट हार्ट सेल आप एक बात समझ क्योंकि जो ड्रग होगा हम लोग कार्डियोवैस्कुलर का हम सब ड्रग्स ही नहीं सकते मतलब हम लोग मतलब रैशनल में बता रहा हूँ कि अगर हमें कोई सेल लाइन चूज़ करनी है तो हमें ये एक तो इसके अलावा भी हम किसी में जैसे अगर हमें इन विवो में कोई किसी ऑर्गन में ज़्यादा इसका मतलब ज़्यादा वो तो उसी सेल लाइन तो इन विवो में मैंने
मतलब क्या चांसेस
उसमें देंगे बीपी की दवा है तो IV तो फिर हम लोग उसके लिए बेस्ट मॉडल ये भी रैशनल बाई फार द बेस्ट मॉडल टू स्टडी
सेल लाइन बाकी जो हम एनिमल मॉडल में ये मैराथन
आप किसी
मेरा भी रिसर्च स्टार्ट होने वाला है बाकी मेरा नहीं आगे चल के साथ ही चीज़ कर पाएंगे
ऐसा कुछ नहीं हमारे सब बराबर है।
नहीं तो मुझे ऐसा लग रहा है हम आइडियाज़ दे सकते हैं प्रेजेंट आप कर दो मुझे आइडियाज़
क्योंकि हमारे
ज़्यादा नहीं करने की जरूरत है कोई प्रीलिमिनरी हमें एक आइडिया है कि हम जो हमारे समझा
Fine. Go to 12, but sit down and leave it.
Nothing is silos, it's all interactive, no? I can't see you together sitting together. Discuss. इसी में तुम पाओगे एक नेचुरल लीडर बन जाता है
इसकी एक्टिविटी इससे सिमिलर है मुझे हाँ तो हम इसमें भी चूज करेंगे जैसे साइबरेशन
वो स्टेप बाय स्टेप फर्स्ट होगा उसके बाद हम उसकी जैसे हम नोटिंग करेंगे उसका बस इससे जस्टिफाई इसमें हम लोग किसी को कोई हार्म नहीं जो मर्जी वो चीज़ और यहाँ हम 5 से
जो मैं बता रहा हूँ चीज़ें बहुत कॉम्प्लेक्स है ईजी
ताकि सबको समझ में
कम से कम
सर जी बोला नहीं
कॉम्प्लेक्सिटी बट 10th फिर ह्यूमन में नहीं
All right, 2 minutes left. We'll be starting with the head presentation. So we'll go in this order: target identification, then preclinical, then clinical. Right?
ये तो समझ माइक लग देना कॉम्प्लिकेटेड तो इसमें एडमिट जो है ना अच्छी आती है.
So just one moment. You can arrange your presentation in such a way what you want to do, why you want to do, and how you want to do, and what do you need for that. So then you ask for funding. What you want to do, why you want to do, and how you want to do. These are the things. And then justify your budget.
SPM सेमी प्रोग्राम में आप रिलीज देखेंगे क्लिनिकल विजुअली अपने डॉक्टर को कितने देर में डाटा जनरेट करेंगे मोस्टली इसमें आप ACR 9 ऑर्गेनाइजेशन चेक करेंगे सेफ्टी टॉक्सिसिटी डोज after preclinical
Yes, I think the target identification, you can come up.
बोलना आपको
इसको ऐसे कर लो ना.
सच ले लीजिए
आप इस पे आइए स्टेज पे. Yeah, can I have your attention now, please? The target identification group. So, just briefly tell your name and then start. The target identification group is going to present.
Assalamu alaikum. I am Madiha Ali Khan and I am going to tell how to do the target identification. So we have chosen breast cancer as our disease. So in breast cancer, first we know that in cancer many genes are overexpressed. So we want to target an overexpressed gene. So first we will go by in silico approach to save our time. So in that we can do like there are databases like GEO database and TCGA database. We will go there and download the data sets of breast cancer, and we'll do the differential gene expression by using R programming or BioPython. Then we will find out the overexpressed genes there, and then after that, when we will find out the overexpressed gene, we can there will be a lot of genes, like 4,000, 5,000 genes we will get. So we want to know one gene. So then we will make the network of those genes, like on the string tool, we can make the network. So we will find like more genes that are more overexpressed, then other genes will be eliminated there. Then we will make sub-networks we can use cytoscape for that. So we will make the sub-networks, and in that there are a lot of algorithms in cytoscape. We can use all of them, and we will find the hub genes from there. Then, for example, we will get one gene that is most overexpressed, that is a top ranked gene, we will get that. And for example, here we can target the HER2 gene that is overexpressed in breast cancer. Then now we want an inhibitor of HER2 gene because it is overexpressed. So for that, we can go on databases like IMPART database that have, from there we can download the plants, the compounds of the plants that can be used for treating the breast cancer. We will make a library of those plants. Then we will do the Lipinski rule testing there by using Swiss ADME, that is a free tool. So there will be a lot of compounds, so we cannot use all of them. So we will filter out the compounds that are less toxic than the other. Then we can go for group docking of those compounds with HER2 gene, and we will found like the top hits compounds. Like if, for example, we get 10 compounds there, then we can do the ADMET analysis of those compounds from Swiss ADME. Then again, so for example, we get 5 top compounds from there that are less toxic than other compounds. Then
So just a pause. So ADME will be coming in the target identification, or it's in the preclinical?
sir, that
Or it's just computational that you're doing it?
sir, yes, that computational part.
Okay.
That is the computational part. Then we can do the MD simulations for finding out what is the best compound. For example, we get one best compound. So here the in silico approach ends here. Then we will go for wet lab, where we can find like a tissue from that is we will do the biopsy tissue that is used in biopsy. So we can get that, and like this part is explained by one of them. Maybe I am not very
Okay,
well in.
good. Any questions, Dr. Ahmed? Yes. Any questions for that?
No, it sounds like a complex.
Good. Yeah. Who's the next one from the target identification? Time is the essence. You have got. I think they have assigned someone to do the next part, right? Come over. The mic is right there. So what we have heard so far is she wants to target HER2. The target is HER2 for breast cancer. The disease that you chose is breast cancer, and the target molecule is HER2. And for that, they have done the computational studies, gene expression studies, and they have also done some ADMET studies computationally. This is the summary.
Okay, so good afternoon, everyone. My name is Riya Singh, and basically I shall be talking about neurodegenerative disease, that is Alzheimer's. So Alzheimer's, it is a kind of disease which causes progressive loss of memory in people, and it ultimately leads to dementia. Now, how we are going to identify the target in this case? See, it has already been well documented that there are 2 proteins which are responsible for causing Alzheimer's. One is beta amyloid and another one is tau. So what happens in this disease is we have normal receptors present on our neurons, and those receptors are called as APP. Now what happens is that there is one enzyme called as BASE1 enzyme, and it tends to cleave that neuron, which leads to accumulation of protein on the brain, and it causes Alzheimer's. So for that, I shall be talking about computational studies which can be used, or in silico studies we can say. So one method is docking. So in docking, what we do is we download the structure of the protein from protein data bank, and after that we tend to minimize the structure of that protein using various softwares that are available, and then we download one ligand of our choice on which we want to do research. So after that we do the interaction between the ligand and the protein, and after that we analyze it. Now after that we use various softwares such as GROMACS and perform molecular dynamic simulations. Now the thing which we lack in our lab in chemistry department is that...
Okay, here don't feel restrictive. Think that you have got unlimited amount of money and everything available. Then what would you do? Don't think with those restrictions in mind.
Okay. So after that we would be using softwares such as GROMACS, and in that we perform molecular dynamic simulations. Now here the thing is, these simulations, they require very high softwares. Like they require GPUs and supercomputers, and these facilities are not available in our department or in the IIT also. So for that we have to send our sample to we call them CDAC, and there it takes immense amount of time, like a month or 1 year. They will analyze that and send the results back to us. So it is causing a hindrance in this research part.
Okay, good. Very good. Let's clap for her. And for the previous one as well. So my question for you is,
Yes,
while you're doing all this target identification, the data that you would be mostly having would be from the computational study. Then you have to show it with the wet science experiment as well.
Yes, wet lab studies.
So
our group, another person is there. She shall be talking
also
about
coming?
it.
I thought there were just two. Okay,
Yes.
just come on, quick then, because we are already 29 here. Come on, we have got just 2 to 3 minutes max. Time is money. Time is of essence. Come on. So you have got 3 minutes to go.
For clinical trials, we do biopsy. We take a normal cell and the disease cell. So in disease cell, the receptors, the biomarkers that are present are overexpressed. Like HER2 receptor, it is generally involved in for repair and the cell growth. So it is overexpressed. So the we can detect. So this cell is disease. So according to that, we develop our drug, like how to deliver the drug on a specific site. Like we can focus on the lock and key mechanism. We develop the drug using the nanocarriers that will go and bind to the site, and then we can do our...
Good. Clapping should be a bit louder. All right, so I would just comment here from the target identification to the target identification group. See, you have to understand the biology of the target. You have to know your molecule and how does it behave. So there are generally some studies done. I'll come there so that you don't have to turn around. So there are some studies that are done which are known as gene silencing studies. So what happens, how to know that Sachin is the batsman who's holding the entire team? By not sending Sachin to play. You will know if the team fails, that means yes, he was the one. So similarly, you do the gene knockout, gene knockdowns. So you silence the gene and see the effect. If yes, HER2 is a target, then if you remove that gene, you will see the removal of that effect as well. So for that, we need to tease out the entire biology and the mechanism. So CRISPR is coming up technology that is allowing us to understand the mechanisms, and we have to understand the entire cascade. Like the story from the this BRAF, the mutations, because this is a MAP kinase, BRAF, the entire pathway, right? So this, unless we know the entire mechanism, we cannot find out the target, that who is coming after what. One's effect can be compensated by the other one. So it's very important to understand it as a systems biology, that about your target, right? Second group.
Just a comment for target identification group, that when you do a target identification, just to tell you guys that when you do a target identification, three things are important. One is the biology validity, that's what he was explaining to you. Second part is druggability, and third part is commercial viability. Okay, so when you present as a target identification, I think you guys have done a tremendous job to tell how we have to do it, fine. But second thing is whether how you will approach, whether it's there you want to do or not to do. So there are three columns you have to make it. Okay, identification, fine, you do some study, you understand whether it's a biology validity is there or not, you think of anything, and then you have to think of, that's the part which you have to take care. The moment you will do identification, then you ask the chemist to start making the compound in the target. Okay, this is how the flows go. So all three things you have to pass on the target. You can't do only identification, say you have to move. Tomorrow you will move and say, oh, it's not druggable, or it is not useful. Nobody will take it. Thank you.
All right, second group. I think who's coming up? Yes, 12:13 already. What, why, how? Yeah. What, why, how, and what do you need? And don't restrict your thought processing by the unavailability of resources. You have got everything. I've got an American next to me. I've got another industry person next to me. They can fund you anytime.
Yeah, sky is the limit.
Hi, Assalamu alaikum. My name is Muzammil, and we have chosen a novel anti-hypertensive drug for our study. So first, we will go for the in vitro studies, and for that we have selected many kinds of assays and protocols. First, we will go for the protein binding, and then before moving on to the in vivo, we will in the in vitro basically we will go for the different cell lines. So regarding that, we will follow the protocol of ADME. In the absorption, we will select, because the anti-hypertensive drug usually they are taken orally, and for that we will go for a cell line which must be relevant to that aspect. So we should select a cell line which is similar to that. For that we will select some cell lines such as CACO2, which is human intestinal epithelial, which is the best for studying the intestinal permeability. And after going through the intestine, most of the drugs usually are metabolized in the liver. So for that, from the perspective of metabolic study, that where the drug goes, how it is metabolized, we will go for some hepatocellular cell lines such as HepG2, which is human hepatocellular carcinoma cell line, which is basically the best cell line for basic metabolism and toxicity studies. And after that, if that drug does well in the in vivo model, so we can also try for from the perspective of clearance from the body, we will go for the renal nephrotoxicity in order to check the nephrotoxicity, we will go for the renal cell lines. For example, we will check its effect in the HK2 human proximal tubular kidney cells, and because many antihypertensive or their metabolites pass through the kidney. So this is one of the aspects which we must focus on. And after we get through all these in vitro assays, and if the drug does well and it shows some positive aspect, then we will move to the in vivo studies. So in the in vivo studies, as per the current literature, so the gold standard in the in vivo studies is held by the rat model, which is best for screening the anti-hypertensive drugs. And before that, we can go for early screening, in which we can use models which are lesser, which are easier to work with from the perspective of ethics. We can, for the early screening, we will maybe see the effect of this drug in the zebrafish, which is best for the optical and heart vessel and genetic studies of this. And after we do the study in the rats, we will move on to the to check its effect in the mice, rabbits, and if it shows positive effect, and it means if we can see that this drug is having anti-hypertensive potential, then we might, before going on to the human trials, we might see its effect in the dogs, because dog is the best for the advanced preclinical stage before going to the human trials. And if it shows positive results, then we will move on to the pigs, which have closest cardiovascular anatomy to the humans, because as we know, in recent times, heart has been transplanted from pig to the human, and that was successful for some time. And we can also use non-human primates, which is difficult from the ethical perspective, but we can do that, and it happens only when the mechanism is highly human specific. If we want to go for further testing, then we can use some primate models as well, such as lemurs or rhesus monkey, somewhat like that. Okay, that's it from my side.
Okay, thank you. I think I've round of applause for very good presentation. So I think you covered a lot of things. You are too much on animal model.
Yes.
You for drug approval, you don't need that many animal model. One question I'm asking you guys: what is missing between you and this group? Target identification and preclinical. What was missing? Lead optimization and assay. They have to provide you the assay. Okay. Then you get a drug. Preclinical start with a molecule. Molecule, you will get the molecule, but target identification people will provide a assay for you. Okay. Lead optimization they don't do the assay. They do the assay, but the whole process is start from the lead optimization, target identification. Okay. Then you mentioned a lot of animals. You will not get anywhere, okay, for that, because animal right people are there outside, okay. They will be having. So for approval, you need maximum three animal model, okay. And for primate, we never used primate. For CNS drug, we have to use primate because we have to see the behavioral effect on that, because they are usually working on brain. Yes. For cardiovascular, sometime it is used. For example, Eliquis, there was no primate was used. I know who developed that. Okay. So this is one thing, okay. Animal model, you start with mice, rat, and in cardiovascular area, rabbit is also another important. You don't have to go to pig or dog even, okay? But primate comes in picture. For preclinical work, there are a couple of things also you have to do, like mutagenesis, okay, AMS test. So before it goes into clinic, you have to do these tests, otherwise at the end, when it goes to market, you will know that, oh, it is mutagenic, withdrawn. So that's done at the preclinical stage in our company. Otherwise, I think anybody else want to present, or everything is okay? Okay, please come. So this was your part.
Thank you so much.
So I am Mehak Sagir. So now I am having a target and a drug. So what I would like to do with the drug and a target, first I will go with the cell line studies, where I would like to study the protein dynamics studies of our drug, if our drug is a protein. So when we do work in the test tube, there are very controlled conditions, but molecular crowding is one of the things that affect the molecular protein dynamics also. So in the cell lines, I would like to do those studies, then followed by the animal models, preferably small one followed by the larger animals. So my aim is to study the efficacy and along with the toxicity of the drug in those animals. So for the toxicity, the retention of the drug in the body, in the preferably the whatever the model we are using of the disease. So retention studies can be done as, I don't know it is done in this way or not, but the PET-based retention studies, where we can study the retention of the drug in the organs of the animal model using very known technetium-99m isotope. The chronic interaction, I am very new to this field, so I would like to study the chronic effect of the drug in the animal model itself, so that we can reduce the chances of failure in the later stage in the clinical trials. Also, organs to be looked for the toxicity. So it depends on the structure of the drug and other factors, that which organs are being affected. As we know that the metabolism of the drug is going to be majorly in the liver, but there may be the chances that the drug may be retained for a longer period of time in other organs also. So I would like to do an essays for those things also. Also the ADME part, so the absorption, distribution, metabolism and elimination. So I would like to go in those essays as well. And I have this thing that when we are talking about the neurotoxicity, so we can have the essays like the permeability of the drug towards the blood-brain barrier. So we can have a replica or a membrane more close to the blood-brain barrier. If the drug is crossing that membrane, we can develop in a laboratory, or maybe the work is going on, I don't know. So it will be easier for us to judge whether a drug is going to be a neurotoxic or not. So it will be more easier and also cost effective. So this is what I would like to do. Thank you.
So there are a couple of things about tissue distribution. You don't use PET reagent. You use radioligand drug. So it will be like a drug is there. We make it as a tritium, which is a radioactive material. And when we dose, this is done at the very later stage in the preclinical side to see the tissue distribution and excretion also. In ADME part also we do that. So PET reagent is for something else. This is.
We use the PET scanner for the scanning. If we attach a radioactive molecule to the drug.
Yeah, but not every molecule is PET reagent. That's what I'm saying. You are making a drug, and I think initially you said you have a protein. You are using a protein as a drug.
So a molecule may be protein.
Molecule may be protein. That's what I hear. That molecule may be protein. That's more difficult now. Okay, making a PET reagent out of protein is practically impossible almost. Okay, so PET reagents are small molecules with a radioligand like lutetium. Coordination. But they are mostly radio labeled. So for doing what you said it right. But in case of small molecule or in peptide, small peptide also, we use tritium or iodine or fluorine. We can replace hydrogen with fluorine, and then we see that it is not, activity is not going down or there is no side effect. Okay? And otherwise it was, that's what I wanted to point out, that otherwise it was okay. Blood-brain barrier, you talk about it, right? So there are many transporters in blood-brain barrier. And there's one assay which is, I think, Dr. Zain, PAMPA is still used here? So PAMPA is the one which is used to simulate, you use the right word, thank you, using simulate the blood-brain barrier. But at the end, you have to do that. It does not mean that you inject to me and take something from my brain. There are many other ways to do that. Okay, thank you guys. A round of applause for number 2. Right.
Let's see who gets the funding, the target identification or...
They already got it. They already got it.
Still they have budget them.
Okay. Destroy कर दिया मैंने $1 billion idea. The language is yours. Oh, Michael, no language barrier.
सर आई एम इंटरेस्टेड इन जो मेरा जो इंटरेस्ट है वो हर्बल मेडिसिन्स में. अभी मैं काम कर रहा हूँ अपने डिपार्टमेंट में, तो मैं नाइजिला सटाइवा एक्सट्रैक्ट पर काम कर रहा हूँ. तो मैंने बात भी करी थी डॉक्टर्स वगैरह से, तो हम नाइजिला सटाइवा का जो एक्सट्रैक्ट है उसको हम चेक करना चाह रहे हैं जो डिजीज़ कंडीशन लाइक एस्थमा और हेपेटाइटिस. तो हमने एस्थमा के लिए बात भी करी थी डॉक्टर से, तो हमने पूरा एक रिसर्च प्रपोजल बनाया भी था, उनको दिखाया भी, लेकिन उसमें बहुत सारी चीज़ें जो अब कोर्स में हमने बहुत सारी चीज़ें जो पढ़ी अब समझ में आई. इतना इजी नहीं है कि हमें क्लिनिकल ट्रायल पे जाना, उसके एफिकेसी को और उसके ड्रग की जो इफेक्टिवनेस है उसको चेक करना इतना इजी नहीं है. उसके लिए बहुत सारा एक पूरा मतलब एक जर्नी है जो हमको मतलब पूरी करनी पड़ेगी. तो उसमें जैसे अगर प्री क्लिनिकल ट्रायल्स में सबसे पहले तो हम चेक करते हैं कि वो कितना इफेक्टिव है प्री क्लिनिकल ट्रायल्स में, लाइक इन विट्रो स्टडीज़, इन विवो स्टडीज़. तो अभी जैसे बहुत सारी रिसर्चेस जो ऑलरेडी हो चुकी हैं उसमें हमने पता करा कि जो इसका नाइजिला सटाइवा का जो एक्सट्रैक्ट है वो इफेक्टिव है. इन विट्रो स्टडीज़ से पता चला कि उसमें एंटीबैक्टीरियल प्रॉपर्टीज़ है. तो उसमें जो एक एक्टिव कंपाउंड है थाइमिक्यूनोन, वो उसके लिए ज़्यादा रेस्पॉन्सिबल है एंटीबैक्टीरियल प्रॉपर्टीज़ के लिए. तो हम उसको एज अ क्लिनिकल ट्रायल में लेकर जा सकते हैं, लेकिन वो इतना ईजी नहीं है, उसमें भी बहुत सारी चीज़ें हैं. अब जैसे फिर इन विवो स्टडीज़ आती हैं, इन विट्रो के बाद इन विवो स्टडीज़ आती हैं. तो अभी तक नाइजिला सटाइवा में इन विवो स्टडीज़ नहीं है, सिर्फ इन विट्रो स्टडीज़ हैं. उसमें एक 2-3 डेटाबेसेस हैं जहाँ से हमें पता चलता है कि ये पर्टिकुलर नाइजिला सटाइवा की जो एक्टिव कंपाउंड है वो क्लिनिकल स्टडीज़ में या इन विवो स्टडीज़ में है या नहीं है. तो उसमें एक आती है कोकेरेन लाइब्रेरी, आती है कोकेरेन लाइब्रेरी, जो हम उस पर विजिट करते हैं तो पता चलता है कि कितने कंपाउंड या फिर कौन सी चीज़ें जो क्लिनिकल ट्रायल्स में चल रही हैं. जब मैंने सर्च करा कोकेरेन लाइब्रेरी में तो उसमें कोई क्लिनिकल ट्रायल में नाइजिला सटाइवा से रिलेटेड कोई भी स्टडी मुझको नहीं मिली. तो अभी तक जो सारी स्टडीज़ हैं वो सिर्फ इन विट्रो स्टडीज़ हैं नाइजिला सटाइवा पर. लेकिन हम ये चाह रहे थे कि हम नाइजिला सटाइवा को हम स्विच करें, मतलब डिजीज़ में चेक कर सकते हैं, क्योंकि नॉर्मली वो लिया जाता है नाइजिला सटाइवा का जो क्रूड एक्सट्रैक्ट है वो नॉर्मली हम डेली लाइफ में लेते हैं, तो वो इतना वो टॉक्सिक नहीं है, उसको लिया जा सकता है. तो हमने एक उस पर रिव्यू भी करा, तो एडीएमटी प्रोफाइल भी करी, तो वो उसमें मतलब कंप्यूटेशनल करी थी, वो सेफ़ है. तो जब हम फॉरवर्ड करें मतलब जैसे इन विट्रो से मतलब हम क्लिनिकल स्टडीज़ में आना चाहते हैं और ट्रायल कंडक्ट करना चाहते हैं, तो उसमें एक तो एडीएमटी प्रोफाइल सबसे इम्पॉर्टेंट है. तो जो एडीएमटी प्रोफाइल है वो मतलब अच्छी होनी चाहिए, एब्जॉर्प्शन, डाइजेशन, एक्सक्रीशन, मेटाबॉलिज्म, टॉक्सिकोलॉजी. तो इसका इतना मतलब अच्छी मतलब एडीएमटी प्रोफाइल तो है. और इसके लिए फिर जैसे क्रूड एक्सट्रैक्ट को हमें लेकर जाना है क्लिनिकल स्टडीज़ में, तो उसमें ये है कि उसमें एक तो फंडिंग का सबसे बड़ा इम्पॉर्टेंट रोल आता है कि जब भी हम मान लीजिए कोई हम अपने किसी क्रूड एक्सट्रैक्ट को एज अ पॉपुलेशन पे चेक करना चाहते हैं, तो फंडिंग सबसे मैटर, जो सबसे इम्पॉर्टेंट रोल है फंडिंग का है. विदाउट फंडिंग यू कैन नॉट डू एनीथिंग. जब तक फंडिंग नहीं है तो हम कुछ नहीं कर पाएंगे. तो उसके लिए मैंने बात भी करी डॉक्टर्स वगैरह से, तो होते जब तक मतलब आपके पास सॉलिड प्रूफ नहीं है तब तक वो रेडी नहीं होंगे, कन्विंस नहीं होंगे वो आपके आइडिया को लेकर। तो पहले आपको पूरा उसके लिए प्रिपेयर होना पड़ेगा। जो सारे उसके प्रोसीजर हैं क्लिनिकल स्टडीज़ से पहले के, वो आपके पूरे फॉर्मेटेड होने चाहिए, वो जो आप उसको अच्छे से उसको प्रपोज कर सकें। तो उसी के लिए रिसर्च प्रपोजल होता है कि आपको एक अच्छे से रिसर्च प्रपोजल लिखना होता है। तो हमने वो भी लिखा, लेकिन इतना अच्छा नहीं था, लेकिन अब ज़्यादा समझ में आया कि उसको किस तरीके से लेकर चलना है उस प्रपोजल को। तो उसमें अब जैसे बताया कि जैसे उसकी डोज होती है, अब एडीमेटी मतलब टॉक्सिक डोज कि उसकी क्या है, अब उसको हमें पता चल गया था 30 mg टॉक्सिक डोज है। तो हमने उसको ट्रायल करा इन विवो स्टडीज़ में, जैसे रोडेंट्स वगैरह, नॉन रोडेंट्स में। तो 30 mg से मतलब टॉक्सिक डोज है, तो हम उसको क्लिनिकल ट्रायल में लेकर जाएंगे, लेकिन उसकी जो डोज वो 1/10 रखेंगे। तो उसको हम 5 mg में लेकर गए। तो इस तरीके पूरा प्रोसेस है क्लिनिकल ट्रायल में। फिर उसको हमें रजिस्टर भी करना होता है, जैसे IND प्रोफाइल करनी होती है। उसके लिए जो इंडिया में जो अथॉरिटीज़ हैं, जैसे DCGI एक अथॉरिटी है, वहाँ प्रोफाइल होगी जो हमारा जो भी प्रपोजल है। उसके बाद फिर DCGI से लेकर और फिर जो जिस हॉस्पिटल में हम कंडक्ट करना चाह रहे हैं ट्रायल, तो वहाँ की एथिकल कमेटी होती है, उससे भी परमिशन लेनी पड़ेगी। तो जब वो उसको अप्रूव करेंगी तो हमें बेसिकली 2 अप्रूवल चाहिए। एक अप्रूवल तो DCGI के थ्रू होना चाहिए और एक अप्रूवल इंस्टीट्यूशनल होना चाहिए, जो गवर्नमेंट जो मेडिकल बॉडीज़ हैं जो उसको परमिट करेंगी कंडक्ट करने के लिए। तो उसके बाद फर्दर हम उसको कैरी ऑन कर सकते हैं। तो इस तरीके से तो आफ्टर 2 मंथ सर मैं बात भी करी थी डिपार्टमेंट वगैरह में, तो हम एज अ उसको नाइजिला सटाइवा एक्सट्रैक्ट को हम उस पर चेक करना चाह रहे हैं एजमेटिक पेशेंट पर, एजमेटिक पेशेंट और हेपेटाइटिस बी। क्योंकि एक्चुअली मैं माइक्रोबायोलॉजी में काम करता हूँ, तो मैंने सर से बात करी थी, तो सर कह रहे थे कि ज़्यादा डिपार्टमेंट का इन्वॉल्वमेंट होगा तो चीज़ें कॉम्प्लेक्स हो जाएंगी। तो हमारे डिपार्टमेंट में हेपेटाइटिस के पेशेंट आते भी हैं, तो हम उसको वहाँ से कैरी ऑन कर सकते हैं। लेकिन सर अभी वो BSc के इतना मतलब एज अ मैं बैचलर स्टूडेंट हूँ अभी, करेंटली आई एम परस्यूइंग माई बैचलर इन मेडिकल लैब साइंस, लेकिन अभी काफ़ी चीज़ें सारी इसमें ऐड ऑन करनी है।
Yeah, you have done a wonderful job, and most importantly, we should also be proud of that we at least given something people understood. And more importantly for you, I will give you some note to do. There is a government Ayush ministry, they are putting money. Your work is much of a related to it. So put a proposal in 2 phases. One is a preclinical, and do some admit study. Your job is good, you have to just prove, and they will ask the question how you are going to the clinical trial if you don't have any data from a preclinical. Of course, there will be a lot of papers and all. If you have that, you can support to it, and then you can get small money, you can start your work. I think that's a wonderful way you can proceed if it has. And coming to that, I think the best part was the identification, and now you touched more that a good part is you went for the tox study. You also told us the MTD also 30 mg, and then you said 1/10 of the dose we will go with the human. That's really wonderful to know that people are understanding where we have to go, and how this is how the clinical trial. Clinical trial is more of a regulatory stuff rather than doing. Important is you know the route. Now you can move. There will be a lot of hiccups, but good job. Thank you. I appreciate your team. You guys have done a good job, you know. You have my number, you have my email. Contact me if you have a proposal. I can at least try to make it better so that people can conceive it, okay? Okay, thank you.